Good morning all, and welcome to Kolpleskaroka, the headquarters of the Papua New Guinea Institute of Medical Research. My name is Dr. Pamela Toliman, and it's my great pleasure this morning to welcome our viewing audience, our research partners, collaborators, and members of the PNGIMR Buttressing Coalition. We have an exciting program planned for this virtual symposium. This is the first time the PNGIMR has delivered a program virtually, but obviously it reflects the season that we're now in. We have many highlights that we want to share with you, the ongoing research that the PNGIMR is undertaking. And I'm particularly excited to introduce to you many of our emerging researchers, scientists and clinicians who work tirelessly to improve the health of Papua New Guineans. We will show you how we partner for research impact. Now, without further ado, it's my great pleasure to welcome here the director of the Institute, Professor Willie Pomat, and also Professor John Kaldor joining us from Australia, who's from the Kirby Institute at UNSW to make their welcome remarks. Thank you.
Good morning, everyone, and welcome to this virtual symposium. Like Pam said, would like to welcome all of you who have an interest in research, who have an interest in research in Papua New Guinea, and who want to be part of the family that we have. So I'd like to welcome all of you. I'd sincerely like to thank John as well for joining, and as well as the KB Institute for being a very powerful partner for us, and all the partners as well. So I hope to see you all throughout the meeting, and welcome everyone. Over to you, John. John, you go.
Yeah, okay, thank you. Morning, Algeta, I'm not sure if that's the right thing to say, but good morning to everybody. And it's a real honor to be able to say a couple of words at the start of this symposium. Just to clarify, I guess the Kirby Institute at the University of New South Wales, where I work, is certainly a key partner of the PNGIMR. But the reason I'm specifically here is I'm alongside Willie, a co-chair of the Buttressing Coalition, which has been in existence for many years, but has really upgraded its activities over the last couple of years to support the endeavours of the PNGIMR and its efforts to improve the health of people in Papua New Guinea. As we have a tradition in Australia now that we always acknowledge the traditional owners of the lands where we are, and that's what I'd like to do first, to acknowledge the owners, the traditional owners in the part of Sydney, as part of New South Wales where I am, which is Arakwal country. But I also like to acknowledge the traditional and long-term owners of all the parts of Papua New Guinea and where our colleagues are based. Because that's the key point about the work we do and the work we collaborate on. It is to serve the health needs of communities and people in these many different parts of the world, but particularly in this stage in our focus on Papua New Guinea and health in Papua New Guinea. The Buttressing Coalition, as I mentioned before, has been in existence for some time, but we had very generous support over the last couple of years from the Australian Government's Department of Foreign Affairs and Trade, which have provided a substantial funding boost, which is intended to match the contributions from partner organisations such as the University of New South Wales Kirby Institute, but also a number of other partner organisations, Burnett Institute, the Telethon Kids Institute, the Walter and Liza Hall Institute, and a number of the institutes that you can all find listed in the Buttressing Coalition documentation. We've been working to support the PNGIMR through the initiatives led by Professor Pomat and the team at the IMR, where needs are indicated, and the Buttressing Coalition is really there just to sort of ensure that support is provided in a timely fashion. And so it's been wonderful to see that evolve, both in terms of infrastructure, but also in terms of supporting training opportunities. And this symposium is a fantastic example of the sorts of development and support that the Buttressing Coalition can provide. So I'm going to say I'm honoured to be co-chairing the Buttressing Coalition and alongside Willie. And we any inputs that you have at any time from both people in Papua New Guinea, but also our partners around the world who are on the call, to let us know how we can do better to support the PNGIMR and its mission of improving the health of people in Papua New Guinea. So with that, I'll hand back to Willie. Thank you very much.
Thank you, John. Obviously, we wanted the Prime Minister to open the meeting, so I guess John and I have, in many ways, just opened the meeting for the audience. And I understand the Prime Minister must be busy with the parliamentary sessions that are currently happening in Port Moresby. So I just only received his executive officer's text this morning to say that she was on compassionate leave and therefore did not answer any of our emails. And so that's so yeah, it looks like we won't have the Prime Minister opening this session, but we will plow on anyway, as we always do with many of these things. So my talk is primarily going to highlight some of the partnerships that have been created over the years, plus some of the highlights of the research that have been done as well. So you get an idea of the scope of work that we're currently doing at the PNGIMR. Next slide, please. So we are the only national public health and medical research in the Pacific region, and probably in many developing countries, we're very proud to be one of those institutions that has a high standing throughout the world. And our current focus is primarily on infectious disease that include malaria, pneumonia, nutrition, sexually transmitted infections, including HIV, diarrhea. And we are looking primarily at the agents that cause this disease, the transmission of them, prevention, the treatment, management, and as well as some of the genetics that go with it, so that we understand what the how the genetics are playing with the host to either enhance the disease or prevent the disease from going ahead. So next. And obviously, a lot of this research is done through partnerships, and we are in the public health and medical arena. So and there's a lot of that here in Papua New Guinea, and therefore our partnership with various institutions globally as well as locally are important in ensuring that the research outcomes help the people that we're trying to help. So, and we are trying to implement the vision of the mission and visions of the Institute over the last 50 years, as well as the partnership is to share knowledge, adapt some of the knowledge, plus to help us build capacity, which include infrastructure, human resources, and research training. Next, please. So some of the research that we have done over the last 50 years, and I'm sorry, I'm only putting only a few of the varied research that we have done. Our studies on pneumonia, for example, started in the 1970s, and it looked at different things, including what are the causes of pneumonia, what are the risk factors, all of those. And from there, it led to the introduction of the pneumococcal conjugate vaccine and the Haemophilus influenzae type B vaccine, which were major pathogens of pneumonia in the highlands of Papua New Guinea. And hopefully we can tell you in the next few years that pneumonia is going down. I know that the immunization rates are not very high, but we can at least say that some pneumonia has declined from the time it was high. We also have studies on lymphatic filariasis, and you can see a picture of that. And from way back, there's a lot of studies happening around what the transmission dynamics were, what the entomology was like, what the causes were, what the worms were, how they looked like, the immunology, and all of that. So that work has culminated in the mass drug administration that is currently being done over in East Wheaton province, which is a pilot province for Papua New Guinea. And malaria, we've also had a lot of work in it. And we continue to work on malaria. Recently, there's been resurgence in malaria, and we found out one of the reasons for why there's been a resurgence, and we continue to do a lot of work on malaria. And Kuru has been one of the long sort of foundation for the Institute. It's been probably the research that has led to the establishment of the Institute. And it's a neurodegenerative disease that occurred in one small part of Papua New Guinea due to the methods of mortuary practices. And this disease has been studied over a long period of time with Professor Michael Alpos, who could be in the audience and who will provide you with a more detailed answers to Kuru itself. But currently we do no longer seeing Kuru in Papua New Guinea. And all of this research, much of it you can see in the PNG Medical Journal, the 99 issues of that journal. Thank you. Next. In the last 5 years, these are some of the research that we've seen. So elephantiasis or lymphatic filariasis, a partnership between National Department of Health, World Health Organization, JICA, PNGIMR has led to this introduction of mass drug administration of ivermectin, albendazole, and DEC in East New Britain. And those 3 drugs we have shown in research in Turkey and also in other parts of the world that it shows a reduction in the disease itself. So that's why there's this pilot program going on in East New Britain. Also up there is the report of high HIV drug resistance, which the Institute also partnered with to find out that yes, first-line drugs for HIV, there's resistance to them, and so we need to change that. And more recently, I think in the last year, we've shown the reason why there's been a resurgence, or at least one of the reasons why there's been resurgence in malaria increases, and that is due to the decreasing bioefficacy of long-lasting treated bed nets. And this is, or was at that time, a politically heavy topic, I suppose, and Global Fund is now onto this. So next. And this is our roadmap for the next 5 years. So we have a corporate plan that begins, that has been endorsed by Council and will help us to guide us into the future. And our vision is to be the center of excellence in public health and medical research, at least in Papua New Guinea and in many developing countries. And the purpose is to lead acceleration of innovations to improve the health and well-being of the population through research and implementation sciences. And there are 5 pillars. And among those, institutional excellence is the first of those pillars. It means that we have to now improve our administrative processes to make sure that the research is supported to its fullest capacity. We continue to also look at the current research activities, and because these are our strengths, and from those strengths, we look at research innovation. What is it that we can innovate out of the current programs, and what can we do for other programs that we are not working on yet? And while doing all of that, we become leaders in the science and evidence-based policy, so that we have our scientists go to various meetings and they're able to provide leadership in the discussions on various diseases. And I guess the last pillar, and probably aptly put last, because it's probably the foundation, is the partnership, because none of this going forward will happen if we do not partner both with local partners and international partners. So one of the partnership areas that always comes up, and one of the probably highlights for us, is research capacity building. And we've got all those young ladies at the top have now received their PhDs and returned to Papua New Guinea. Dr. Janet Garwe was the first woman PhD for the Institute of Medical Research, so we congratulate her for that. Dr. Moses Laman, who's now the Deputy Director, and unfortunately is on compassionate leave at the moment, but he was going to be doing the next part of this talk. And we not only build our research scientists, but also our administrative staff. So at the bottom there, you can see a number of our drivers who we try to ensure that they get trained in first aid, so that if they are doing pregnancy-related studies, for example, or they're involved in various accidents and so on, at least they have some first aid ability. And on the far right at the bottom there, you can see a new drawing of a building. And we hope to build this new infrastructure in Madang province very soon, because at the present time, all the good research that for malaria and lymphatic filariasis that come out of Madang are conducted through infrastructure that was built in the 60s and it's deteriorating, and we're spending a lot of money. So we're waiting for, in partnership with the government, hopefully that we can have this building built starting this year. Thank you. Obviously, we also partner with governments, and the government of Papua New Guinea itself, the Australian government through Centre for Health Security and DFAT. And one of the biggest partnerships is the trilateral program, which looks at malaria diagnostic, and it's a partnership between the government of Papua New Guinea, Australia, and China, and that's looking at diagnosis of malaria. So it's a partnership model that is an example for many other programs to follow. We also, there are also donor agencies like Global Fund, World Health Organization, NIH, of course, and NHMRC and others that also we put in competitive research grants that we're able to then conduct research in Papua New Guinea. So we are very pleased to partner with governments and donor agencies of those governments as well. Obviously, all our research is community-based, and so we ensure that our partnership with communities important, and ensuring that our staff who go out there have a good rapport with all the communities that we go and work in, so that we can get the best data from the communities to also help the communities, and also have the communities themselves help themselves to better their own lives. And so the different partnerships that I've spoken about through the talk, I guess the Buttressing Coalition is a culmination of all of that, because it's a platform for partners and friends to come together. And it started back in 1999. Professor Michael Alpers was then the director at the Institute, and began the idea that we should have a Buttressing Coalition to support the Institute, not only in the science, but also in other areas that are important to ensuring that the science continues well. And so in 2019, at the 50th anniversary in 2018, we began discussion on changing the administration of some changes to the Buttressing Coalition. And in 2019, membership fee structure was put in place, and this is managed through the University of New South Wales, and with agreement through the Indo-Pacific Center for Health Security, who committed 2/3 of the funding. If the members are able to contribute 1/3, they'll provide 2/3 of the funding. So the 7 members currently at the KB Institute, Burnett, James Cook University, Swiss Tropical Public Health Institute in Switzerland, the Melbourne University, Walter and Eliza Holt, West Farmers, and West Farmers Center for Vaccine and Infectious Disease in Perth. And we hope this year we add others into the Buttressing Coalition. And the funds that come out from the Buttressing Coalition have so far allowed us to upgrade IT. So that's why I'm able to talk to you virtually without meeting many of you. But that's the improvements that we're currently seeing. We've renovated buildings in Port Moresby, as well as provided small grants for our scientists to study COVID-19, which is the current pandemic. In 2021, we are hoping to build renovations in Mark, support this virtual symposium, and also to develop, have a consultancy to develop a process to improve employer benefits. So, and hopefully we get, like I said earlier, some more partners to join the Buttressing Coalition. So now I'll go into some of the research that are highlights of what the Institute has done and are currently doing. And before I do that, I'd like to get you through the structures. So the Institute reports directly to the Minister of Health, and that is through the council. So we have a 7-member council that helps us to ensure that we are going along the right path. And I, as a director, also support the council to ensure that the administration goes along the path that the council has for us. And under the deputy director, there's 2 deputy directors. One of the deputy directors is for science and research, and that's Dr. Moses Laman. And there are 4 units that are currently in place: infection immunity, sexual and reproductive health, population health and demography, and vector-borne disease unit. So in each of those units below them, you can see the different diseases that are studied by those units. And the studies of those diseases encompass clinical, epidemiological, laboratory, behavioral, and health systems, as well as surveillance and monitoring and evaluation. So beginning with vector-borne, and I know this is a very busy slide and we won't go through it, but a lot of the ongoing work at the moment is looking at surveillance. So the STRIVE PNG network, for example, is funded through Center for Health Security, and it's a surveillance looking at malaria, in 8 sites around the country, and ensuring that we use a software called Tupaya to see if we can get real-time reports of where malaria is and find out whether responses can happen. And also nested into that is the funding through ACR, looking at Japanese encephalitis and as a zoonotic disease. So with the help from CSIRO. And there are also other studies on malaria currently ongoing. One of those, at the bottom there, you can see the NatNat study. NatNat obviously is mosquito in dog fishing, and it's studying the new vector methods that we are currently studying, looking at to see what other vector methods, what other methods to reduce mosquitoes and so on. And that's currently being held in done in Medang. Obviously, we talked about lymphatic filariasis. And in the pipeline, we're hopefully looking at establishing operational research, looking at meal lab equipment with partners in Switzerland and Korea to ensure that to look at technology currently available that can read slide microscopy instead of humans. The Taunam study is also about mosquito nets and also is related to the NatNat study, which are looking at vector control programs. The next unit is sexual reproductive health unit. And like the previous program, there's quite a lot. And the primary program for sexual and reproductive health is around the clinical aspects as well as the behavioral aspects. And you can see health-seeking behaviors for TB, as well as HPV, viral load. And the clinical aspects look at HPV point-of-care testing, as well as the one-time trial, which is funded through Wellcome Trust, is looking at women's health, the birth outcomes of women who might have sexually transmitted infections. And the ECTAP program for HIV, looking at HIV drug resistance, is also funded through Center for Health Security, and is looking at HIV drug resistance in Papua New Guinea. Obviously, as this year, we've got a couple of COVID-19 grants as well through WHO, as well as through small grants. So those COVID-19 are evaluating antigen RDTs, COVID-19 serology, and also COVID-19 impact on health services. In infection immunity, we're looking at different studies on pneumonia and also looking at gut microbiome and determinants of host, severe host determinants that determine childhood pneumonia and what genetic factors might be influencing pneumonia, making some kids much more susceptible to pneumonia than others. And also part of that is looking at the genomics and epigenetics and all the omics related to vaccination. And so this study has started and is a collaboration between the Institute and the Boston Children's Hospital. And it's primarily looking at whether vaccines, what sort of pathways, the metabolomic pathways, when you give a vaccine, they enhance or suppress so that you get a protective immune response or suppressed immune response to various diseases. Obviously, polio surveillance has been conducted and completed, and we continue to conduct influenza and rotavirus surveillance. And the team is also looking at responsible for COVID-19 diagnosis for the country. In the pipeline, we're looking at environmental salmonella typhi, as well as hopefully we look at surveillance of meningitis. The population health and demography unit has two major studies, or three. The CHESS program is a longitudinal health and demography surveillance in 4 locations in each of the regions of Papua New Guinea. And they also help to conduct the government's WASH program and a program called the Sunrise, which is looking at child movement. And the CHESS program is funded through the government of Papua New Guinea, through the Department of National Planning. The trilateral malaria, as I introduced earlier, is a program between the government of Papua New Guinea, Australia, and China, and it's looking at the diagnostics. And obviously, trilateral malaria sits between population health and vector-borne disease unit. We also conduct major malaria indicator surveys for the country, looking at the malaria program that have been implemented to determine how much malaria there is. And at the maps at the bottom, you can see that over time, we saw a decline in malaria cases, and then there was a resurgence in 2017. And one of the reasons I highlighted earlier was the lack of efficacy in bed nets that were being used. So a lot of these programs you will get to hear in the spotlight sessions later today. And what's in the pipeline are wasting and stunting, which we are hoping to conduct a study in Eastern Britain with the Bennett Institute that's currently going through ethics. And also nutrition in the Wanang area of Madang province with St. Louis, University of Washington in St. Louis, I think. So, and hopefully, that helps us understand why people in that, or health services in those areas, what the problems are and what nutrition they can help themselves with. There are other studies that are not necessarily packed under any of the different units, and these are the Healthy Mothers, Healthy Babies, which we conduct with the Bennett Institute in Kokopo, in Eastern Britain. The ACE entity, which is conducted through John and his team at the University of New South Wales, and we hope to nest some of this into the current mass drug administration studies that are currently going on in Eastern West, will happen in West New Britain and are currently happening in East New Britain. The Fleming Fund is a national government support for national government for laboratories throughout Papua New Guinea, and we're supporting that as an SNS advisory institution. Also, in looking at the antimicrobial in the Fleming Fund, we also have the AMR ARC hub through the KB Institute. The TrueNet is looking at TB through the Bennett Institute. And the surface studies also part of the program we are conducting with Sussex University in the Wanang area of Madang province. And like I said earlier, we are conducting COVID-19 diagnosis for the country here at the Institute. So we have 2 sites that are conducting testing, one here in Goroka and also in Port Moresby. And to date, we've sampled almost 30,000 samples that have come through, and of that 1,154 have been positive. On the table below that, you'll see that in 2020, around each of the different quarters, we're seeing a rise in the number of samples coming in and the number of tests that were conducted. In 2021, you'll see that in Q3, we haven't done very much yet, and in Q2 as well. There hasn't been, the numbers are not as high because primarily because some of our reagents have run out, and there's been global supply issues that also affect our testing. And so we're seeing that people who are coming for tests are the ones who are most likely to have a symptom. One of our major problems is turnaround time, and I guess that's the main reason why the government has now shifted from from ensuring that all symptomatic cases are tested using GeneXpert or RDT, and that all the contact tracing counts as PCR. So at the bottom there, you can see that a lot of the testing now is decentralized to provinces. Each of the different provinces through the TB program that was funded through Global Fund, each have GeneXpert machines, and these have been adapted for COVID-19 testing. Thank you. And these are some of the research, COVID-19 research that are funded through the Buttressing Coalition. And the young scientists on the left, young in terms of science experience, because some of them are, I guess, not so young. And we anticipate a lot of this research to be conducted soon. And you can see for yourself the different, the biomedical side of research that will be conducted by some of our scientists. And these are the social behavioral research that will also be conducted by some of our scientists as well. And we hope to complete the 11 research activities by the beginning of next year, so that we can, at the next virtual symposium, we can all sit down and go through some of the results that our scientists have put together. So thank you very much, everyone. Obviously, throughout that, we see that partnership and collaborations at all levels, from the community all the way up to global networks, are all important for the Institute to integrate the results and to make sure that the outcomes are good for our people. We look forward to new collaborations, both national and international, and we thank all of you for taking part in this program, so that you can also be part of the network that helps us to conduct research to improve lives of our people. Thank you very much.
Thanks, Professor Pomat, for that great introduction to the partnerships that we have. And we're going to move to our first spotlight and have a listen to John Paul Matlam, one of our health extension officers who's doing a lot of research and actually the clinical lead for our COVID-19 work at the IMR.
Hello everyone, my name is John Paul Matlam and I am a research clinician with the PNG Institute of Medical Research. I've been with the Institute for 7 years, or a little over 7 years now, performing different duties from, managing the clinic to going out to the field to collect data and samples. I think I'd run out of time if I were to list down all the opportunities for career growth that I've come across while working at the Institute. But I think my current role as the lead clinician for our COVID-19 response has given me the opportunity to work with a variety of people, not just in the health-related field, but people from your CEOs to the normal person on the street. And each of these experiences have had its own life lessons. What am I passionate about? I would say I am passionate about seeing positive outcomes, and especially in the health-related field, I have a particular interest in public health, and particularly in identifying and formulating ways to make healthcare more accessible to the rural and remote areas. I personally believe that as a clinician with further training in microbiology, I am more aware of the need for evidence-based medicine, especially when it comes to antimicrobial and antibiotic therapy. I am better able to advocate for antimicrobial stewardship and also give advice to my peers. In the next 5 to 10 years, I'd like to see IMR more involved in the training of health personnel, because I believe research is an important component and students need to have that foundation in order to perform better in the workforce by identifying the need for proper collection, recording and analysis of data, and seeing how these influence policies. So my advice to students wanting to pursue a career in research is to go for it. Because if you want to see change, you need to know how to provide research-based evidence to influence outcomes.
We're now going to move on to our first session, which is on eliminating lymphatic filariasis. This will be led by Professor Chris King from the Case Western Reserve University in the US.
My name is Griffin Tabun and I work as the project manager for the East New Britain Neuendorf project. I will be talking to you a little bit about what I do, my experience in the field, and what I've learned along the way, and overall what is involved in the running of this project. The objective of the DOLF project is to develop and test improved treatment for onchocerciasis and lymphatic filariasis that will enhance efforts to control and eliminate neglected tropical diseases. In Papua New Guinea, the focus is on eliminating lymphatic filariasis, which is the endemic disease in this country, through mass drug administration, otherwise known as MDA, of antifilarial drugs. And that includes DEC, albendazole, and ivermectin, in combination known as the triple drug therapy, or IDA. So this was conducted in East New Britain, which is highlighted in red on the map. It has a population of more than 300,000 people with 4 districts, and prior convenience sampling showed East New Britain to be endemic for LF. So the drug was distributed across the whole province and treated more than 300,000 people, which had a 91% drug coverage, which is really good, considering that this was the first MDA conducted on a provincial scale as compared to community level scales in other parts of the country. So prior to MDA, we conducted baseline monitoring and evaluation of filariasis infection across 49 villages in the 4 districts. And then in 2020, we had a 1-year follow-up across 48 villages. And our results showed significant reduction of LF 1 year post-MDA across all the districts, except 2 hotspot sites previously known to have very high infection. However, the very low infection all across the whole province showed the success of the MDA program after a year. Coordinating a large project can be demanding but rewarding nonetheless. And the success behind the project involves manpower, cooperation, and resources. And the experience in managing or conducting the work well in the field can go as planned, or it may not go as planned. So despite planning well, nothing can deter unexpected events like bad weather, accidents, sickness of staff, lack of cooperation, and other eventful situations. But I consider them all learning curves to readjust, refocus, continue the work until completion. So here are a few photos of the work in the field. You can see that much of the areas in the province is challenging to access. So we see our cars crossing flooded rivers. Some of those places do not have vehicles for transportation. So our health staff have to carry the equipment and walk from one village to another. And also the boats, some of those locations cannot be accessed by road. So the boats are required to transport people from one place to another. Again, we see how village people or the community help in transporting or carrying some of the things from one site to another. And we see some workers setting up their station beside the sea to collect samples in each of those communities. And some of the mishaps or eventful situations that we've actually experienced is, unfortunately, one of our car vehicles got swept away by a river current during a flood. And you see some of our vehicles, or one of our vehicles got bogged down in the mud because our survey forms got soaked when one of our vehicles got into the water. We brought the surveys form back into the office, dried them up, and then took them back into the field to collect the data. So there's a lot of things that happen behind the scene that many may not be aware of. But we all see the data, but we don't see the work that goes into it. So I give credit to my team that actually are able to go there, endure the challenge, and come back and we see the results of their work. So the blood-in monitoring surveys involve blood surveys. Monitoring evaluation involves blood surveys, and that includes testing people for circulating filarial antigen or CFA to detect the antigen of the adult ones. And then if the screening is tested positive, then blood samples are collected for microfilariate screening. And that is usually conducted at 9 p.m. because the parasite is nocturnal and only circulates in the bloodstream during the time. Some of the problems that we encounter is that participants can live without having the blood collected because it's nighttime, they want to go to bed, they're tired, to wait around, and also bad weather, which prevents people from participating. And also field supplies can run out. There may be shortage of some of the kits. And so this disrupts the work while they're in the field. However, despite all of this, the team, the field workers, they are resilient, they work through adversity, and they conduct the research in the field, they collect the sample, they store the sample, they bring them back into the office, they help with processing and data entry. So really they are the backbone of this project, and I give them credit for their work and the effort in it. And I look forward to working with the team for future projects that is conducted in East New Britain and probably elsewhere in the country.
Hi, I'm Lincoln Timino. I'm currently a senior scientific officer based at the PNGIMR Medding branch, and I will be talking to you about some of our experiences with xenomonitoring of lymphatic filariasis in Medding. Now, the mosquitoes used in the xenomonitoring are taken from the human landing catch method, where an individual sits outside of their house with an aspirator, a torch, a clock, and with 6 cups, and collects mosquitoes between 6 p.m. and 12 a.m. on an hourly basis. And we have up to 17 collectors per village. And we do 2 checks at 8 p.m. and at 10 p.m. to make sure the collections are done correctly, and then that collectors are not sleeping. The cups are brought to us at 12 a.m. in the respective bags. Now, some of the challenges that we faced, apart from walking around in the night and getting attacked by dogs, is getting the collectors to start on time. And also when a collector has to leave for another appointment immediately, and we had to select someone from the house and train them quickly so that they can continue collecting mosquitoes at that spot. Now, once the bags of cups have been brought to us, we then go back to our base where we set up and start to process the mosquitoes. And we separate the mosquitoes according to their species and according to their feeding status, whether they're fully fed, partially fed, or unfed. And then we store them in Eppendorf tubes and store them in 9x9 boxes. The first night is usually a struggle, but then the proceeding night it becomes easier. Now, the main challenge was to enter the data directly into the tablet in real time, but then we got the hang of it, and the data is usually uploaded once it has been entered. Being able to upload the data directly into a cloud server made our work really easy when we're back in the lab, where we can just download the data and select the type of samples we want to process, and then we pull out the samples, do DNA extractions, and then we perform WB-PCR on them and run them through a gel electrophoresis to see if they have the Wuchereria bancrofti filarial worm or not. And it was quite interesting for us to see the drastic drop to 0 mosquitoes carrying the filarial worm after an year of MDA. And even 2 years after the MDA rollout, there were still no mosquitoes with the filarial worm. Thank you.
Hello, my name is Chris King. I'm a professor of pathology, and I've been working in Papua New Guinea now for almost 30 years. And I'm sorry, apologize for some of the technical difficulties. That's my fault for not getting on Zoom. But anyway, I wanted to talk a little bit of a title of the talk and introduce this lymphatic filariasis in Papua New Guinea. And I titled it Punching Above Its Weight because Papua New Guinea has had a huge impact on our approach to lymphatic filariasis. So the next slide, please. Okay, here we go. I got it here. So just quickly on lymphatic filariasis, and some of you know a lot about lymphatic filariasis, so I apologize for that. But for those who are a little less familiar, Papua New Guinea has about 5.6 million people at risk of filariasis. 14 of 22 provinces are still endemic. And Papua New Guinea has some of the heaviest burdens of filariasis in the world. And what we know about filariasis is that Willie showed a few pictures before, but here are some of the pictures just from the last year or two of work in Eastern Britain province. You can see it's a disease of the lymphatics, causing lymphatic dysfunction, resulting in really quite profound swelling and abnormalities in some of the limbs that could be quite disabling. So this is really quite a burden, certainly for those people who have it, and the infection rates are quite high. So this is a big issue there. So what we know about filariasis, many people know it, it's a mosquito transmitted disease. The Anopheline mosquito is the mosquito that transmits. It's the same one as malaria in Papua New Guinea. So that's important. It's a fairly inefficiently transmitted disease. And that's one of the reasons that it's been elected to be targeted for global elimination programs. And also the major cause of lymphatic filariasis is caused by Wucheria bancrofti, which doesn't have any, is not a zoonotic or other reservoir. So if you can eliminate in humans, you can eliminate the vast majority of lymphatic filariasis worldwide. So again, it's a vector borne. The adults live in humans. They have a reproductive time of adult males, maybe about 5 years. And there are several drugs that are very active against the parasite, particularly DEC, ivermectin, and albendazole that work at different stages of the parasites. So what has started is approximately about 25, 30 years ago, an effort was made to set up a program to eliminate lymphatic filariasis. And this is a program because it's endemic in many countries, 80 countries, in fact, when they started looking at this in the 2000, with 1.3 billion people at risk of filariasis and probably 120, 130 people known to be infected. So a program was developed to eliminate lymphatic filariasis called Global Program to Eliminate Lymphatic Filariasis. And this has been very successful and is based upon giving doses of drugs and mass drug administration I'll talk about shortly. And what's going on is that these drugs have been given, well, close to 8 billion doses have been given, and it's resulted in a significant decrease in the countries that are endemic for filariasis and has resulted in elimination in some countries and eliminated as a public health problem. But still, a lot of people are still at risk. So what I'm going to talk to you is a short story about PNGIMR's role in this program and how they played such a critical role in doing this. So if you see in the next slide, I'll just show you is that this is sort of how this program is designed to eliminate lymphatic filariasis. And this is probably the largest, arguably the largest mass drug program for treatment of any infectious disease ever attempted. So it's a huge undertaking involving many countries. And basically, I won't go into a lot of details. It has two components. One is the mass drug administration to eliminate the transmission of lymphatic filariasis. The second part, the lower part, is control of the morbidity. Now that's something we haven't really done a lot here in Papua New Guinea, is the morbidity control, but really the focus has been elimination by interruption of transmission. And how this is done is working on, as I've highlighted in these circles, is areas where the PNGIMR research has had a critical role in supporting and providing the evidence to support this strategy. So the vector control or integrated vector management has been key in reducing the exposure of people, obviously, to the stage of the parasites. The other part is showing that mass drug administration, if given to enough people and successfully, repeatedly can actually eliminate the transmissible stage of the parasite and result in interruption of transmission, which is necessary to go on and do elimination. And so this is sort of the approach. That's what's going on in Papua New Guinea and other parts of the world. And there's been successful elimination in many countries in the Western Pacific, but Papua New Guinea still is needing to do this. So I want to highlight that this thing is really done by partnerships, as was brought out by Willie, and these are so absolutely essential. It's really a combination of the global program to eliminate filariasis with the national programs, working with the WHO, the pharmacies, donate the drugs, and the different donors that are important for supporting everything from, as you can see, a number of listed, and this is not a complete list at all, other academic organizations working with JICA and other non-governmental organizations have been all critical to be successful in moving this thing forward. PNGIMR has been really critical for the operational research. So what are the key studies, and I'm just going to say very highlightly, that have actually been seminal studies that have supported this program? So it all started probably back in studies back in the 1990s. And this is work, a publication by a number of people that are on the calls or have worked with Papua New Guinea IMR to show that mass drug treatment given repeatedly can interrupt LF transmission. And this is one of the important studies to support that approach for elimination. It was done back in the 1990s and follow on 2000s. The next thing is that they showed that if you can interrupt transmission, bed nets were critical. And these insecticidal treatment bed nets is shown to reduce transmission. So vector control is very important. If it's integrated with the mass drug treatment, you can have success. And more recently, and some of the people have already talked about that, is better treatments for elimination of filariasis. And Willie already mentioned is this triple drug regimen that was started and started originally in Papua New Guinea and has now been adopted worldwide for many areas, not every areas, for treatment of lymphatic filariasis. So certainly this has been a huge impact of these three types of studies and just many more. I'm just highlighting these that have had impact on filariasis here in Papua New Guinea. So what are the current plans for LF elimination in Papua New Guinea? Well, as I mentioned, there's 14 provinces that are endemic. These are the things that get treated. New Ireland province, which just had mass drug administration. And as we speak, they're organizing trips to determine whether transmission has been interrupted in New Ireland province. As Willie has mentioned, we've done one round of mass drug administration with IDA, interrupted by COVID, and we're planning hopefully to do the second round this year, if we're lucky, if things go. And this is going to be some of the first studies to really show how many rounds are going to be successful in doing it and what would it take to interrupt transmission using many fewer rounds. West New Britain province is planned for going on forward and East Sepik province after that. So those are the current plans of moving through these provinces and doing elimination. And I just want to highlight this, as Willie also mentioned, is that the integration of these things is really important. And as we bring in more partners, is that the global program really creates a strong platform of drugs that can be mass distributed and monitoring evaluation. But this can also have an impact on other infections. For example, scabies or soil transmitted helminths. And as we broaden these kinds of treatment and deliveries, we can have impact of the bed nets on malaria and other investigations on leprosy on the morbidity management. So a very important part moving forward is not just LF, but integrating these with other kinds of projects. So basically, I'm sorry, we just had, these are some of the people I've asked, or we've asked to give a little highlights on it. Krafinta Bun just talk about her experience in the triple drug treatment in East New Britain province. And then we'll have Nelly Sanuku talking a little bit about her experiences with the triple drug therapy in Moprik, who sort of led that, and Lincoln to talk a little bit about experience of monitoring the transmission by looking at the mosquitoes. Thank you very much.
Hi, Chris. I wondered if you could hang on for a sec so we could ask you a quick question.
Sure.
We have a question that's just come up in the chat function. We're wondering if there's been any impact on mosquito numbers following the triple drug therapy with ivermectin.
We haven't really looked at that systematically. My suspicion that the ivermectin given in the doses that are given probably have relatively little impact on mosquito numbers, because the doses and the duration of the sufficient levels to have an impact on the mosquitoes is probably only 4 or 5 days. I think where you're really going to see an impact are people using higher doses or more frequent administrations. But no, we have not looked at that in a systematic way. other people have and have not found a big impact in other areas. Thank you for the question.
Great.
Hi everyone, my presentation is about my involvement with the lymphatic filariasis studies that were carried out here in Drakiki, Isipik Province, Papua New Guinea. I'm Nalisa, a clinician registered with the PNG Medical Board as a health extension officer, and I'm currently employed with the PNG Institute of Medical Research, working as a research clinician, a lab manager, and a certified dangerous goods officer. I've been working for almost 13 years now. I was initially employed as a research clinician and a team leader with the lymphatic filariasis project here in Drakiki, Isipik Province, and I've been working with the project for close to 10 years. My role with the lymphatic filariasis project was leading a team of 7 staff doing field research on human population in high LF endemic communities in Drakiki. My role includes setting up of work schedules, giving community awareness on the LF disease and the studies that will be carried out, and making sure the information is clear and well understood by the study participants. Doing diagnostic survey using fingerprint blood samples to identify LF infections in individuals in the community, doing written informed consent on individual who meets the inclusion criteria for the study, processing biological samples in the lab to further check for microfilarial intensity, anemia, abnormal biochemistry, doing urine analysis for pregnancy and other abnormalities, as well as giving the results back to the individuals and referring those with abnormal findings to the nearest health facilities with the logistic support from PNGIMR. Storing and shipping of biological samples to other designated IMR laboratories, and also help in analyzing data and reporting to the study coordinators and principal investigators. Being a female officer and a new mom back then, leading the team doing field research was very challenging for me, but I was able to manage and made sure all LF studies were completed and the study goals were achieved. The 2 studies that we did that have great significant outcome on the LF treatment guideline in the world today were the safety and pharmacokinetics of single-dose triple drug therapy with diethylcarbamazine, albendazole plus ivermectin. It was done on 24 participants and went through for a total of 3 years, which includes the years of preparation, follow-ups, and the finalizing of the results. The second study was the efficacy study of the single-dose triple drug therapy with diethylcarbamazine, albendazole plus ivermectin versus the standard drug therapy, which is the diethylcarbamazine and albendazole. It was a randomized clinical trial that was done for 24 months on 182 participants. Overall, it took a total of 5 years to complete the study, which includes the years of preparation, follow-ups, and the finalizing of the results. The results of both studies have shown that the single-dose triple drug therapy with diethylcarbamazine, albendazole plus ivermectin is safe and effective in clearing out the microfilarial in the blood and sterilizing adult worms until they reach their expected lifespan, of which is 5 to 7 years, as well as reducing the cost of logistics to provide for repeated rounds of mass drug administration with the standard drug therapy with DC and albendazole for 5 to 7 years, the adult filarial worm's lifespan. We were able to complete the study with these proven results through funding from external funders and Case Western Reserve University in collaboration with PNG Institute of Medical Research. And with the proven results, the World Health Organization has approved the single-dose triple drug therapy to be used worldwide in LF endemic areas outside of sub-Saharan Africa. I am pleased that with the approval by the World Health Organization, my people, the people of Papua New Guinea, who have been living with and stigmatized due to the disfiguring, neglected tropical disease, will now have treatment made available in all health facilities throughout the country. Thank you. I am also pleased to be involved with the LF studies that were carried out, and not only here in Drekiki, Isipik province, but also in Bolgna, Madang province, and East New Britain province, Papua New Guinea. Thank you. My acknowledgement to the people who have helped me make this video presentation, Noel Berry and Ann Lasson. Thank you so much.
Okay, this is Chris King again. Thank you very much for listening to the pre-recorded sessions, but I wanted to open up to ask any questions. I understand that Patricia Graves had a question. Patricia, do you want to ask that question?
What was Patricia's?
Chat. Oh, but she must be in the chat.
Chris, you'll have to read the question.
The problem is I have the chat for the Zoom. Yeah, because I think the chat comes up in the web and I had to close that. Yeah. Okay, so anyway, as that comes through, as we hear the chat, well, thank you very much for listening to this. I hope you saw things. I think it's an exciting time, I think, in Papua New Guinea. I think it's a tremendous opportunity for us to be able to move forward with the opportunities for doing LF elimination. I think the successes that have been occurring in New Ireland province, and more recently with a good success in East New Britain provinces, more donors have come on board. For example, I just learned last week that the USAID is now about ready to support money to help with the elimination program in Papua New Guinea. So I think that's a big move forward along with WHO. So I think there's some real good opportunities for that. So anyway, I think here we go.
I've got the question. In the
Yeah.
surveys in East New Britain, any systematic collection on lymphedema or hydrocele burden?
Well, we did. I would say that we have some of this information available on when they did the mass drug administrations on doing that. We have to analyze those data and putting it forward. In the surveys that we did with the monitoring and evaluation, we absolutely did survey that issues. And of the close to 5,000-6,000, we found it very focal, like in a couple of villages, and I showed you examples of people in villages, burdens were very high, like we saw maybe 10% of the people had evidence of lymphedema or other things. Other villages that had lower levels of burden, we found relatively little disease. So it's highly focal. But when it's there, it's there. You saw some of the examples at the beginning. Good question, thank you.
Well, excellent. Well, Chris, we just wanted to thank you so much for leading this first session. It was an excellent introduction and just setting the scene for what's current and happening in lymphatic filariasis. Thank you for your continued partnership, friendship to the Institute and for always helping us out.
No, my pleasure. It's a privilege for me to be able to work with such a great group of folks. Thank you.
Fantastic. We're now going to move on to our second spotlight, which is on Dr. Nalisa Neuendorf, who's a social scientist and senior research fellow as well. And after her spotlight, we're going to go into our second session, which is a pre-recorded panel discussion on point of care test and treat innovations. Thank you.
Hi, I'm Dr. Nalisa Neuendorf. I'm a research fellow at the PNGIMR, and I'm going to be talking today about social and behavioural research in public health in Papua New Guinea. As a trained anthropologist, social anthropologist, there is a lot of importance in what social and behavioural research can do for public health. In the work that I have done, it focuses a lot on qualitative methods. The research work that we do within our unit is both quantitative and qualitative, but today I'll be talking a little bit about qualitative methods and the importance of words and stories. So qualitative methods, in my mind, and certainly from my experience, helps to enrich and enliven the voice of participant lived experiences. It breathes life into numbers and data. It shows that we are more than just players in the stories of our own lives. We produce stories to make sense of the world that we live in. And in these moments of creating stories, we make claims, we create and we reinforce knowledge that tells us something about our society, it tells us something about our culture and collective, and it also tells us something about ourselves as well. So what is the impact of social research in public health? It can provide detail and context, it can provide a point of view, it can provide conceptual frameworks of analysis. And for the researcher, it involves reflexivity and critical reflection of the method and process of research and data collection, which is very important to strengthening and enhancing what is done in the future. What else can social science do, and what has it done? Sexual and Reproductive Health Unit have an extensive list of projects that use social science to enhance public health. One such example is the work with people living with HIV and the point of care testing. And social science has been able to produce insights to help health service provision. Social science can also identify strengths in local community health promotion, in advocacy and social justice initiatives, supporting gender-specific programming. And I've certainly seen this in my short time over the last 2 years working on several projects, and also in partnership with ongoing collaborators at James Cook University and Pacific Adventist University to support sexual and reproductive health and well-being in PNG. Social science makes sense of the lived experience, and I understand this through undertaking a PhD in which I collected stories from people who witness violence happening in their community, and through their witness stories are able to explain and detail and make sense of what that violence means to them, and also then explain the type of response they believe is appropriate to the violence that they witness. So future impact of the work that I am doing involves social research and social media, particularly COVID-19 and the sharing of health information, and also using more creative and visual methods such as photo voice to in the COVID-19 and health service provision work that I'm currently undertaking. I thank you very much for the time, and that's all from me.
Good morning, everyone. As Deputy Head of Sexual and Reproductive Health Unit, I'm honored to open our session today on the partnerships and innovation in point-of-care testing and treatment to improve sexual and reproductive health in PNG and other high-burden settings. In the last 10 years, the PNG Institute of Medical Research has led a collaborative research program in HIV, sexual and reproductive health, and have worked in partnership with national and provincial health authorities, clinic experts, and medical societies, and communities and patient groups to pioneer a number of world-class innovations, co-designed, evaluated, and are translating into practice in PNG. Notably are the following: point-of-care HPV testing and same-day treatment, and for cervical screening, first introduced by our team in 2013, has been shown in 2 large-scale field trials involving more than 5,000 women in PNG to be highly effective, acceptable, cost-effective, and feasible to implement in routine health care settings. This approach was recently endorsed in the new WHO screening guidelines and is set to be scaled up in the Western Pacific by our collaborative group as part of the new WHO agenda on cervical cancer elimination. Secondly, the point-of-care HIV viral load testing was first introduced to PNG in 2016 by our team as part of the County Me Too Biobehavioral Survey. We were first to implement such a survey with 9 different point-of-care tests, including viral load, urogenital, and anorectic sexually transmitted infections, and TB. Our team has been able to produce some of the first HIV cascades of care for key populations globally. Now we are using point-of-care viral load to provide routine monitoring of the development of HIV drug resistance. Then we have the point-of-care early infant diagnosis for HIV in children less than 18 months, which was field trial in PNG in Myanmar and was shown to be highly acceptable and feasible to implement in routine care health settings. It also showed POC testing enabled more timely initiation of ART for our children in PNG. In addition, the PNG Institute of Medical Research is a leading world first to evaluate point-of-care testing and treatment of STIs in pregnancies to improve birth outcomes, the one-time trial. That is due to complete in the late 2021 and expected to report findings in early 2022. Today, as we meet, our teams are actively involved in partnerships, bringing these technologies to eliminate cervical cancer and to diagnose and monitor HIV as part of routine service delivery in several provincial hospitals. Finally, I would like to thank Associate Professor Angela Kelly-Hanku and Professor Andrew Vallely, who in their joint positions with PNGIMR and the Kirby Institute have jointly led our collaborative research program in sexual and reproductive health in PNG for a decade and a half, and who continue to deliver, actively co-lead, guide, and support our work in translating innovations into real-world benefits for the people of Papua New Guinea while building the capacity of our team. I now want to warmly welcome my colleague, Dr. Pamela Toliman, who herself did her PhD as part of our program of research on HPV, to lead a panel with 5 of our partners with whom we are collaborating these world-first innovations. They will each highlight it is not just what we do, but how we do it. Thank you.
Today's panel discussion will focus on point-of-care test and treat innovations in Papua New Guinea. And it's my great privilege to welcome and introduce to you today's panel members.
They are Dr. Paulus Ripper from the Western Highlands Provincial Health Authority, Dr. Peniel Boaz from the National Department of Health, Janet Sangopa from IGAT Hope, Dr. John Bolgna from Odilon Hospital in Madang, and Caroline Hanau from the PNG Cancer Foundation. Welcome, everyone.
I might start our panel discussion today with a question to Dr. Paulus Ripper, who is the Director of Clinical Standards at Western Highlands Provincial Health Authority. Dr. Ripper, the PNGIMR has had a strong and long-standing relationship with the Western Highlands PHA, and recently this was cemented with the signing of a formal MOA. In your opinion, how has this partnership benefited the PHA and Western Highlands province as a whole? And do you have any advice for other PHAs who would like to partner in health research activities?
Thank you. I would like to take this opportunity to thank PNGIMR for giving me an opportunity to express my gratitude on behalf of the people of Western Highlands and the Western Highlands PHA for that robust partnership we've shared over the last few years. It has, with the several programs going on, given us a head start where we are implementing the point of care testing already for women for HPV and children for early infant diagnosis. So it has been a very good partnership for both of us. Now, in terms of, and I'll refer particularly to the HPV and viral load testing, which are the programs that we are partnering at the moment. Now, in Western Islands and Juwaka, I'm from the local communities where almost everyone has lost a female relative to cervical cancer. And now several thousand women have already undergone testing, and many express relief after testing when they know that they are free from the scourge of cervical cancer for a few more years. So from a personal viewpoint, that has had a large impact on women in my communities. From a clinician's point of view, it reduces the hopelessness that one feels when we diagnose a woman with cervical cancer, because from here on, it's not early enough to save lives. It usually goes on to be fatal. As a PHA, we know now that we can begin to go out into the communities and offer the programs that allow preventative and early interventions to the population we serve, rather than waiting in hospitals for the sad and futile inevitable. With the early infant diagnosis program as a pediatrician, I felt the hopelessness of parents who did not know the status of their babies whilst they were waiting for the results of the DBS test that was sent to Port Moresby, and those were taking a turnaround time of several months. Together with viral load testing, we can now offer these patients a lot of relief, and the design of having a one-stop shop for patients with HIV, for instance, which can improve patient satisfaction and compliance, and the viral load testing, which will go a very large way towards meeting the goals that we have set nationally for HIV, the 95 goal. It has given the management of the Western Islands PHA the impetus to begin to review how we manage the overall HIV program in the province. So overall, it has been really great for us to work together with PNGIMR. My advice to other PHAs is to proactively engage with PNGIMR. And this must begin at the level of the management, with the CEO taking an active role in managing that partnership. The PHA must have a shared vision with PNGIMR to utilize the research findings to improve healthcare to the population they serve. Whilst doing that, I think on a practical level, the PHA must have a focused person with whom the research team can engage with, so that the inevitable logistic difficulties that arise at times can easily be sorted out for both organizations. I think that's all I can sort of say at the moment, but it's been great to work with this vibrant PNGIMR team that visits us, and with whom we work together.
Thank you, Dr. Ripa, and we certainly, it's certainly a privilege for us to have this relationship with the Western Highlands PHA. Partnership working and harnessing innovation to improve public health indicators are key themes of today's symposium.
Dr. Peniel Boaz is the HIV and STI Program Manager at the Department of Health, the National Department of Health. Dr. Boaz, would you share with us today your thoughts on how a close working relationship with PNGIMR has enabled such innovations to be translated into practice in Papua New Guinea?
The National Department of Health with PNGIMR have a very strong partnership and in working collaboration for many years. PNGIMR has provided many supports to the National Department of Health programs, and this support includes research, capacity building, trainings, technical support in terms of clinical as well as laboratory support. And I acknowledge all the support from PNGIMR in collaboration with the University of New South Wales as well as the Kirby Institute. In terms of the research component, PNGIMR has worked closely with the STI program in terms of researchers in STIs as well as HIV. And many more research were conducted, I would say, in the last 20 to 30 years. And that has been very beneficial in terms of advising the program itself moving forward in terms of policy development and revision of guidelines as well as strategy. Last 5 years or so, we have been able to revise our STI guidelines as well as our HIV guidelines, depending on those researches. Just highlighting some of those research that we've done with support from PNGIMR is the gonococcal sensitivity research as well as the HIV drug resistance studies, as well as the IBBS studies. And those are the big highlights in terms of helping the program moving forward, in terms of moving forward in having new innovations in terms of improving the quality of care. We have worked with PNGIMR in terms of using the new platforms such as the Gene Expert platform, providing quality care by the point of care for viral load testing as well as EID testing project. And we've started working on that in one of the biggest sites in the country, and this Western Islands province. IMR as well is a strong member to the technical working group in the STI program, and has contributed a lot in making sure data and statistics supported through by the evidence-based research that they've provided. So thank you so much for giving me this opportunity to share the experience with PNGIMR. Thank you.
Thank you, Dr. Boaz. IGAT Hope has been a key PNGIMR partner for many years. Janet Sagopa, who joins us today, is the president of this national organisation and network of people living with HIV. Janet, you have been a key advocate of rights and responsibilities of people living with HIV, including their right to self-determination and participation in decision-making that affects their lives. What has a partnership between PNGIMR and IGAT Hope meant for yourself and also the community that you represent?
What has the partnership between PNGIMR and IGAT Hope meant for myself and the community? I will speak from the perspective of the partnership between PNGIMR and IGAT Hope and the community we live contributes to the capacity building of people with HIV and HIV health services in PNG through community health training, clinical mentorship, advocacy, and governance support. The PNGIMR also provides a comprehensive program for people with HIV and affected by HIV, so that can benefit from learning and also the latest the techniques and the research methods. This is also, I believe that there is a cooperative relationship where we agree to share responsibilities, specific achieving specific goals. Such as the research that was done by the IBBS survey, which they are the members, we are the key populations together with, and the Count Me In, the human rights, public speaking, and the HIV drug resistance viral law. The partnership also enhanced communities to contribute to the AIDS response in many different ways. The leadership and the advocacy ensures that the response remains relevant and is grounded. Also keeping people at the center, and then there's no one left behind. The communities that I'm referring to include the peer workers or the peer educators, the network of people living with HIV or affected by HIV. This includes such as a gay man, or men with sex with men, women and young children, counselors, community workers, other doctor step service providers, and civil society organizations and the grassroots activists. Again, this software is an important platform to highlight the roles and what we've done in the communities at the time when, when there's reduced funding and a shrinking pace of the civil society, putting the sustainable services and advocacy efforts in jeopardy. Also, say there's a really we have this other strong partnership. The strong partnership work with the PNGIMR and IGADO. So I've said earlier that we were involved in the IBBS survey and Count Me In and the human rights as well. And we really acknowledge and appreciate the work that PNGIMR is doing towards keeping the people with HIV intact and making them to understand and fully enhance with the knowledge of what is HIV, living with HIV, and can continue living. I think that's all I can have to say for now. I acknowledge IMR for the great effort and the great work that we work together with the GADOP, and we look forward to working with you more in the future, in the years to come. Thank you.
Thank you, Janet. Thank you. We certainly appreciate this partnership that we've had with IGAT Hope, and congratulations on the incredible work that you continue to do for your community. Dr. John Bolgna is a senior obstetrician and gynecologist and head of department at Modilon Hospital in Madang. Dr. Bolgna, you have worked with PNGIMR now for many years, and you've played a lead role in the fight against cervical cancer in the country, and also a key role in the research on new solutions for screening and treatment of women in Papua New Guinea. How has your close relationship with the PNGIMR and the community in Madang enabled these innovations to be translated into practice?
Thank you, Pamela. I have had 11 years of long cordial relationship with PNG Institute of Medical Research, with research collaborations and partnering in clinical services provisions here in Madang, and also with partners in dialogue with Goroka. During this time, I have been involved with several big research collaborations and clinical trials, which have actually changed clinical practice guidelines in Papua New Guinea. and other resource-limited settings as well. As we know, the burden of cervical cancer in Papua New Guinea is immense, and the government has no proper comprehensive clinical strategies for primary prevention to tertiary care involving radiotherapy. At the moment, it is quite fragmented, and everywhere else in the country, they're doing things on ad hoc basis. In Medang, realizing these challenges, we embarked on fundraising drive in the community to build infrastructure and have other necessary equipment and instrument in place for possible HPV point of care testing and treatment, which we had and knew was working. The community of Medang has been very supportive. Some of the fundraising activities we did include the Color Run, the Medang Beauty Pageant, and climbing Mount William, which is about 4,500 meters above sea level in Simbu province. And recently, the Mount Kanagio in Karar Island in Medang. Those funds raised, we were able to build infrastructure and have other instrument and equipment in place. And when the infrastructure was set, I had to ask Professor Andrew Vallely that I was ready if we can have this HPV point of care testing and treatment established here in Medang. And he was very kind with others to have agreed, and we set the HPV point of care testing and treatment here in Medang in 2018. And so far, we've done really well. We now have an efficient clinical referral pathway system that is very comprehensive, and we know that it is working. We know it is hard to convert research work into sustainable clinical services provisions for our local people. And so we are working really hard to secure funding to sustain these services with involvement of other friends locally, as well as other international partners and friends. For instance, Cabrino Hospital in Victoria has been able to support us in sustaining the clinic with some grants, which I and Andrew are working on at the moment. So it would be really nice for local PHAs and the government of Papua New Guinea to actually take ownership of these programs, which we know is actually working, and such as the HPV and point of care testing and treatment, so that we can really eliminate the burden of cervical cancer in this country. And I look forward to any future collaborations and partnering to extend our services into the rural and remote parts of Medang, as well as Papua New Guinea, where the majority, almost 85% of our people live, particularly the vulnerable population, which are women. Thank you.
Thank you, Dr. Bolgna, for all the incredible work you do for the women of Papua New Guinea. Point of care HPV testing and treatment for cervical cancer has been pioneered in Papua New Guinea. This approach was recently endorsed in new WHO guidelines, and it's set to be scaled up in the Western Pacific region as part of WHO's agenda on cervical cancer elimination. Caroline Hanau is a cervical cancer survivor, a patient advocate and board member of the Papua New Guinea Cancer Foundation. Caroline, what impact do you think a scaled up new approach to cervical screening will have on women's lives in Papua New Guinea and the Pacific as a whole?
Thank you, Pamela. Thanks for inviting me to be on this panel. Just with regards to the scale up, I think it's extremely important. Without this service coming into PNG, it's very limited. It'll be very limited for women, as we know already. This service being available to women in PNG and within the Pacific, as we already know, there's already a limited service. There's nothing really available for women, particularly in the rural areas, in terms of screening. So with the scale up of this service, it would just bring so much more confidence, I think, to women to know that there is something available for them, that they don't need to fear testing, that it's something that's viable, it's feasible, and it's something that they'll be able to come into town and do, and know that they can have their testing done and have their results within a few hours is so important. From my perspective, when I was diagnosed, it was really hard to figure out where I could go from there just to find out what testing was available, what happens after that testing procedure. So I'm really excited to think that something like this in the very near future is available for our women, particularly, like I said, in the rural setting where it's most vital for them to know that they have a service. A lot of the time, it's something that's considered taboo, having any sort of gynecological screening. And I think it's important for women to know that they can come and have it done safely and securely, and to know that there is a support service there for them moving forward. So it is, again, a vital service and extremely important. And it also saves, I think, that cost of knowing that they can go somewhere locally and not having to think about the costs of, having the test done abroad, or having to think about traveling abroad, which is a financial burden, it's an emotional burden. It's something that the whole family takes on board with anyone that's diagnosed at any particular time. I think from my perspective as a cancer survivor, one thing that was really challenging for me going through my journey is I know how blessed I was to be able to go overseas and have treatment. As blessed as I was, it was also, as I've mentioned before, a financial burden and an emotional burden. So I really feel it's important as researchers come on board, we really need to have the government support for women to know, firstly and foremost, that the service is available. Cancer screening and services are available in Papua New Guinea. Secondly, they need to feel confident that we can be treated here. It's really hard to imagine women in rural areas just feeling so alone and so helpless, not knowing what to do and where to go. I think it's really important and I think it's vital for the government to come on board, and it really is important for the women of this country, especially in our rural areas and rural settings, just to know that there is services here for them that's available to them, that they're comfortable with, that there is somebody here in country that's here to support them and help them through this journey, because it's difficult. It really needs the government's support and the government's willingness to help the women of this country. And that one thing I always say is that our homes are not anything without the women in them. And for so many women to be dying of this every day is just, we need to do more. I think Papua New Guineans in general deserve more. And there should be less of us dying of cervical cancer in PNG.
Thank you, Caroline. What a great way to end our panel discussion today. Thank you very much for tuning in.
Well, hope you enjoyed the pre-recorded panel discussion that we had with some of our most valuable partners in the space of point of care, test and treat innovations in Papua New Guinea. We certainly wouldn't have achieved what we have without these valuable partners in both at the PHA level, in the community, advocacy groups, as well as the National Department of Health. We had a question in the chat function regarding HPV vaccination and what is happening in terms of progress on that to date. HPV vaccination is a crucial component of any elimination strategy, and that's certainly something that Papua New Guinea needs to consider, and PNGIMR is working towards, with development partners, to work toward that end. So that concludes the end of our session 2. Thank you. I hope you enjoyed the session and found it interesting and informative, challenging also. We are going to break for 10 minutes. Please use this opportunity to stretch your legs, get a cup of water, a cup of coffee, and we will see you back in 10 minutes' time. Thank you. welcome back everyone to this virtual symposium by the PNG Institute of Medical Research on Partnerships for Research Impact. We're going to move on to our next spotlight, and that was on Dr. Gwendolyn Vengiau, who is a Senior Research Fellow in Public Health.
I never thought I would engage or be interested in nutrition until I embarked on my master's research project that required me to identify a disease of importance in my country so I could develop a research proposal. This led me to realize the change in disease pattern in the country, as shown in the table from the Global Burden of Disease. As you can see, there's rising NCDs, and infectious diseases continue to be a problem, apart from malaria, as you all know has shown a positive outcome. Then I asked what could cause the shift, and the literature pointed out that nutrition was one of the major contributing factors to this change in disease pattern. And this is when I began my work in nutrition in PNG. Our work in PNG indicates that the country's nutrition situation is indeed the shift from traditional to more modern diet, and how people were adapting to this change, as well as coping with food insecurity. Though we continue to have our fresh vegetables, we're now starting to see that there's more foods such as fried flour. And this is now common in many villages across PNG. This tells us that adverse diets are now reaching the vast population in the rural area. Our research also demonstrates that adverse dietary changes include use of oil, salt, and most importantly, sugar intake from sweetened beverages. And these dietary changes vary across population in the country. Therefore, it is vital for us to document and understand these adverse dietary changes so we can develop and implement effective public health measures to address this rapidly emerging issue. Also, whilst undertaking this research, it highlighted the importance to develop right methodology and suitable tools to collect this essential data so we could provide accurate evidence or information to contribute to policies and legislations that will protect the people of Papua New Guinea in relation to nutrition. Another realization was the significance of obtaining the skills and knowledge to analyze and interpret dietary data, which many a times is sent overseas for analysis and interpretation. Given the importance of nutrition and having the expertise, I hope to link nutrition research with other important scientific research work that will contribute to improve the health of our people. And this could be achieved through collaboration. I also believe that we should work across institutions such as agriculture and education, so we could enhance information that will assist in understanding the issues that are impacting the health of our people. Thank you.
Thanks. We're about to go into our third session for today, but I just wanted to acknowledge all the audience that's logged in and tuned in and is participating via the chat function and tuning into what we're sharing via this virtual symposium. And our last check, we had 84 viewers on, and we do know that some of those are actually many people, multiple people watching from one login. So welcome and thank you so much for joining us and participating in this event. Our next session is on accelerating the control of malaria through research partnerships, and this will be led by Dr. Maria Ome-Cayus, who is a senior research clinician.
Thank you, MC, and hello everyone. My name is Maria Ome-Cayus, and I am a senior research clinician in the Vector-Borne Disease Unit, which is led by Dr. Moses Laman. Vector-Borne Disease Unit will be leading this session, which is about accelerating the control of malaria through research partnerships. Malaria has been a major research focus for PNGIMR for over 45 years. Through the years, the Institute had collaborated with many partners from both within PNG and from abroad who continue to collaborate with the Institute in malaria research. Research into malaria and other mosquito-borne diseases such as lymphatic filariasis and arboviruses are primarily conducted by the Vector-Borne Disease Unit. Vector-Borne Disease Unit is perhaps the biggest research unit in IMR, comprising almost half of the Institute's workforce. Up to 80% of this workforce are employed on research project grants, the grants that are jointly won by researchers from IMR and from partner institutions. Vector-Borne Disease Unit has strong technical expertise in epidemiology, clinical trials, microscopy, entomology, molecular diagnostics, and immunology. And in this 25-minute session, we hope to showcase some of these core functions of the Vector-Borne Disease Unit in malaria research and the partnerships that allow the research programs to generate new knowledge and evidence to improve their health outcomes, both in PNG and globally. Let's now move to our first presentation.
This presentation will be focused on using a partnership-based approach in implementation research with support from a coordinating project management unit. This is the outline of the presentation. The first part will be led by Mr. Leo Makita, and he will focus on the partnership approach of the project and its guiding principles, and I will lead the second part and focus more on the role of the project management unit.
Different settings have different definition of partnerships. What we use under Strive and Trilateral is focused on genuine partnership where everyone contributes from their place of strength and every partner feels valued. This slide reflects the partnership continuum. One extreme reflects a partnership governed by money, while the other extreme is focused on co-created activities and everyone's involvement. This is not to say that one is better than the other. It highlights different perspectives. Having defined the kind of partnership that we utilize under Strive, Trilateral, and other projects under Vector-Borne Disease Unit, you may ask, how do we do this? This approach is guided by what we call guiding principles. As reflected on this slide, these principles guide how partners behave. They would include building common purpose along a shared journey, promoting mutual benefit through knowledge of vulnerability as well as strengths, promoting equity and voice across within partners, and promoting ownership and sustainability within PNG institutions, harnessing diversity across partners, promoting transparency, fostering equity, and finally flexibility and adaptness.
Partners feel valued when we identify their strengths and utilize their strengths. The analogy of this car summarizes what we mean by this. Different partners bring experience, skill set, status, cultural influence, expertise, settings, etc. All this adds value to the project and ensures and guides how the project moves forward. To ensure that all aspects of the project function in a coordinated manner, the project management unit is the coordinating body of the project. They ensure that project operations are always ongoing. The governance structure of the project provides guidance on how project management unit operates. There are different groups that the PMU or project management unit engage with for different things, starting from decision making to implementation of the activities. Thank you.
Hi everyone, my name is Henson Dima and I work as a project laboratory manager with the Papua New Guinea Institute of Medical Research attached with the vector-borne disease unit for the last 10 years. I will be speaking about the challenges faced out in the field and how our work on malaria research over the years and the partnership established with the communities around Papua New Guinea are utilized to overcome those challenges. As we know, research is very important for evidence-informed decision-making and policy change. Our work on malaria research to support evidence-informed decision and the fight against malaria has taken us out into some of the remotest part of Papua New Guinea, where basic healthcare and other vital services are of serious concern. Reaching out and helping to provide basic healthcare and malaria treatment are often very challenging. In this short presentation, I will highlight some of these challenges faced and how partnership formed over years with communities, health facilities, and other stakeholders play a key role in overcoming those challenges. Now, the challenges, I have grouped them into 4 groups. Operational challenges, staff. In Papua New Guinea, when you employ someone, we say you employ a whole community, meaning the staff carries the burden of the community to work, and this can affect their performance. For instance, if there is a death in the community, it is his or her cultural obligation to be there to support the family or community. Thus, this can affect his availability at workplace. Power outage, transportation issue, communication are also common operational challenges faced on a daily basis. Community engagement, low literacy level. As we are aware, much of Papua New Guinea's rural populations have education up till primary level, or some do not at all, which can be very challenging when communicating the research purpose and methods. This can lead to misconception about IMR's work and thus develop unwanted expectations beyond our capacity to control. Unexpected movements and relocation of participants. Papua New Guineans are very mobile. They can get up one morning and decide to relocate to a different community, district, or province for days, weeks, or even years. That makes it very challenging, especially for follow-up and data collection. Logistical challenges, poor infrastructure development, bad road and bridges associated with uncertain weather conditions such as heavy rain and flood can be of problem when reaching out into rural communities. Most communities are not connected by road and are only reachable by foot or boat. This presents very challenging conditions for research work. Traffic issue is also a common issue when traveling frequently out to reach out to the community. Law and order issues such as alcohol and drug abuse, petty crimes such as holdups and community fights are also common problems faced out in the field that can hinder the operation or progress of your project. The lesson learned is that fieldwork strengthens an expertise. Unexpected changes and challenges occur which may demand immediate decision and action based on local knowledge and experience. Having worked for over 45 years on malaria research, PNGIMR has this network and a connection to address those challenges. This enables PNGIMR and its partners to conduct research collectively in partnership with communities to improve the health, especially for those participating communities and PNG at large. Now, the importance of partnership, long trusted relationships. With our long-standing work across PNG, we have established relationships and trusted networks within communities that enable us and our partners to carry out successful malaria research and data collection that provides a platform for informed decision-making at different levels of health. Whilst in the process of conducting research, we are also able to work with our partners and communities to try to assist with the much needed healthcare needs for the community. Local knowledge and experiences. The experience and knowledge we gain from the partnership about the different challenges is shared among the community and different stakeholders to strengthen and successfully carry out the work of malaria research and also better prepare us and our partners and the community of the new project work that is coming into the communities. Thank you very much.
Hello, my name is Tamara Kuliala, and I am a scientific officer with the Vector-Borne Diseases Unit based in Port Moresby. I will be sharing with you the laboratory capacity we have across our sites for our vector-borne diseases projects, particularly for malaria. PNGIMR's vector-borne diseases laboratories are set up across 3 provinces. We have laboratory work established in Medellin, Port Moresby, and Maprik in the East Sepik province. Maprik is a small site that has coordinated many studies that have contributed to local and global policy changes for vector-borne illnesses. The laboratory capacity of Maprik comprises of a microscopy unit and a specimen preparation laboratory. Malaria, lymphatic filariasis, and soil-transmitted helminth infections have been studied with techniques like the CATO-CATs for helminth infections established in Maprik. The Medellin site is located within the Yagao Rural Hospital facility. We have laboratories spread out across the site, as you can see on the slide. Our microscopy unit has skilled level 1 microscopists. Our entomology team is the largest on site, and we have the entomology lab and field teams working on projects that focus on understanding wild and lab-bred mosquitoes and how they spread vector-borne diseases. Our immunology lab looks into understanding how we respond to malaria, and our molecular parasitology laboratory focuses on understanding these diseases and causes through genomics. Our partnership in health laboratory in Port Moresby has facilities that can support immunology, molecular, and diagnostic studies for multiple infectious diseases. We are based in UPNG's School of Medicine and Health Sciences, Tarama campus. This lab is part of the PNG Molecular Hub, which includes partners from UPNG, the Central Public Health Laboratory, and NACIA. We have pooled our resources for molecular testing, which enabled the facilities to support diagnostic testing for the recent emergencies of COVID and more recently, African swine fever. The PNG Molecular Hub supported PNG's COVID-19 response through both molecular diagnosis and serosurveillance for samples collected from all provinces across PNG. Prior to alternate molecular assays becoming available, like the GeneXpert, the hub supported the response through running almost 10,000 PCRs. Support from project partners in our malaria projects were very helpful in enabling us to support this response. Our Port Moresby and Medellin sites have the capacity to do molecular testing through real-time and conventional PCR. For serology work, we have established ELISA setups in both our sites. We also have the MagPix device, which allows us to study antibody responses to multiple antigens. Our Medellin laboratory has been involved in research and development activities through field evaluation studies of prototype devices. An example is this rotating magneto-optical device, where it basically uses magnets and lasers to detect malaria using the magnetic properties. Our laboratory capacity has enabled us to be involved in various projects over decades. These are some of our current projects. All of our projects involve partners from universities and institutes, both here and abroad. The ISMR project comprises of a global network of independent research centers in malaria endemic settings, which were established to provide the knowledge, tools, and evidence-based strategies that's crucial to understanding, controlling, and ultimately preventing malaria. It has been ongoing for almost 10 years now. The Trilateral Malaria Project was established through a trilateral agreement between the governments of PNG, Australia, and China, and aims to improve malaria diagnosis through partnerships of all responsible organizations in PNG with support from abroad in China and Australia. The STRIVE PNG project carries out integrated febrile illness, molecular, and vector surveillance at 8 sentinel sites across PNG. The Trilateral Malaria Project and the STRIVE project also support medical sciences students through research projects. Our other projects look into various aspects of malaria, from drug studies, epidemiological studies, and molecular diagnosis of malaria and other viruses. We also use our facilities to support monitoring and evaluation for the National Department of Health programs like the Mass Drug Administration and the Malaria Control Program. We hope to become a center of excellence in the region, expanding into the space of neglected tropical diseases. We are planning to improve facilities to support genomics, particularly sequencing in country to inform public health programs. All of this is possible through partnerships. We value the relationships through international and domestic partnerships that have supported capacity building for local researchers and our facilities. Despite the challenges, it remains possible to collaboratively conduct highly relevant research studies in Papua New Guinea. Thank you.
Hi, I'm Lincoln Timinau, currently a senior scientific officer based at the PNGIMR Meding branch, and I will be providing an overview of the ongoing work at the entomology laboratory here in Medang. The Ento Lab comprises of the largest team under the vector-borne disease unit, and we have currently 28 staffs working at the entomology lab, and our work involves both the lab and the field. Now in the lab we have an insectary, which is the only one in the country, where we rear Anopheles faraudi mosquitoes, a common malaria vector in PNG, with the capacity to produce over 3,000 female mosquitoes per week. Now these mosquitoes have been colonized for over 50 years and are quite susceptible to any form of insecticides. Hence, we have been using these mosquitoes to test the bioefficacy of long-lasting insecticidal nets using the WHO cone bioassay. We have also been quite successful in infecting these colony mosquitoes with Plasmodium vivax, which makes us one of the few labs in the world that can actually do this, because it is quite difficult to grow Plasmodium vivax in the lab. Hence, we need access to infected individuals. Now, this puts us in a very unique position where we can be able to collaborate with other institutions to test potential transmission blocking vaccines or antimalarials that can inhibit transmission. We also have staff who are experts in performing the WHO bottle assay and the WHO tube assay with wild-caught mosquitoes. And we also have an ELISA machine that we use to detect sporozoites in our field-caught mosquitoes, and also PCR machines to confirm the morphological ID of our mosquitoes that are caught in the field. Now, as part of our research into understanding the biting patterns of malaria vectors, we collect mosquitoes overnight using the human landing catch method and the various screens. We also do larval collections to understand the ecology of the mosquitoes. And we have staff who are experts in performing morphological IDs out in the field or when the mosquitoes are brought back into the lab. Now, apart from focusing on malaria vectors, we also look at other vectors such as the vectors for arboviruses, where we are currently setting up traps for. Now, because of our laboratory setup and the type of work that we can do, we have been able to collaborate successfully both locally as well as internationally to carry out research over the last 2 decades, not only into malaria vectors but other vectors as well. Now here is a list of some of our ongoing studies. Now the ISEMA and the STRIVE PNG have been discussed by Tamara. And now the ACIAR ZEPA is essentially a collaboration between the Australian Centre for International Agriculture's Research Zoonotic Arbovirus Project and PNGIMR with NACIA as well. And the main aim here is to establish environmental surveillance of vertebrate hosts and mosquito vectors of zoonotic arboviruses as well. Now, the DOLPH project is a global effort to eliminate onchocerciasis and lymphatic filariasis using the triple drug regimen. Now, the triple drug regimen is currently being rolled out in East New Britain, and we are assisting with providing the xeno monitoring. Now, the Lihir Island Malaria Elimination Project is an initiative of the Newcrest Mining, which is based on the island, and it is aimed at ridding the island of malaria, and we provide a vectoral surveillance support. We are also involved in a countrywide insecticide-resistant monitoring for the National Malaria Control Program. Now, the two other new projects that we are currently involved in, the NatNat and the Taunam, will be further discussed by Maria. I would like to highlight here one of our work where we partnered with the Rotarians Against Malaria and the National Malaria Control Program that led to an immediate change. Now, in this study, we noted that LLINs distributed after 2012 did not meet the WHO criteria according to the WHO cone bioassay. Now, this resulted in an immediate change in the LLIN procurement strategy in PNG. And now PNGIMR is at the helm of monitoring the new nets that are being distributed. Now, this work wouldn't have been possible without the help and support of not only the Rotarians Against Malaria and the National Malaria Control Program, but the Department of Health, the University of Liverpool for performing the confirmatory tests on the nets, and James Cook University. Thank you.
Thank you, Lincoln, and hi again, everyone. As you have now learned from my colleagues, malaria research conducted at IMR by the Vector-Borne Disease Unit is always a collective effort from various individuals, institutions, and countries, and that collaborative approach to research can be mutually beneficial and maximizes impact. Working collaboratively is particularly important in a country like PNG, where resources are limited. As highlighted earlier, through partnerships, partners can participate in joint decision-making, they can pool and share resources, and can maximize impact of evidence in influencing policy. For instance, IMR has been monitoring and evaluating the impact of the National Malaria Control Program interventions for the past 10 years, providing updated evidence that can guide malaria control programs. Through the international partnerships, Vector-Borne Disease Unit is able to source assistance, resources, and expertise that we may lack here in PNG. Such partnerships have also been instrumental in building and setting up infrastructure and equipment to allow IMR to undertake high-quality malaria research that can inform malaria and health-related policies here in PNG and elsewhere. Capacity building has always been a priority at PNGIMR, and these partnerships provide opportunities for training. Included here on the slide are faces of some of the individuals who have graduated or are currently in training for Masters and PhD, and many more have done their honors. IMR not only builds capacity within the institute, but through partnership also contributes to the capacity building in the partner institutions. This is just one example of such partnerships. Here, the students from the School of Medicine and Health Sciences use IMR laboratory capacity to generate data that is needed for their different projects. Through that, IMR provides training in laboratory and research methodologies, as well as provide mentoring and supervision. Overall, PNGIMR strives to undertake innovative, high-quality research programs that are relevant and aligned to the country's health needs and those that can have significant impact on the health of the people of the country. Before I finish, I want to introduce a few of the program of work that Vector-Borne Disease Unit at IMR will be leading in the coming months to years, with a focus on evidence to inform policy. NatNat, which stands for Newly Adopted Tools, Network Against Mosquito-Borne Disease Transmission, is a large project that will be conducted by IMR and various partners, as shown on this slide. Currently, long-lasting insecticide-treated nets are the only vector control tools that are programmatically implemented against malaria in PNG. Vector biology such as the early and outdoor mosquito biting and the mosquito's preference for a variety of habitats are indications for the need of additional vector control tools. But again, there is the lack of infrastructure. This work will therefore establish the capacity at PNGIMR to test the additional vector control tools, including indoor residue spraying, larva source management, and spatial repellents. The core objectives of this project is to strengthen the capacity to test these new vector control tools in PNG, conduct evaluations of these tools, investigate acceptability and cost effectiveness of these tools, and finally to support a national malaria control program-led formal network for vector control tools and interventions in PNG. If these tools are found to efficiently reduce malaria transmission, they could be integrated into the National Malaria Control Program. The second program of work is PAVE, or Partnership in Vivax Elimination, which will be conducted by PNGIMR and many other partners as shown on this slide. PVivax poses a major challenge to malaria control efforts. Its unique biological ability to lie dormant in the liver cells as hypnozoids allows it to escape detection and treatment. These hypnozoids then become the source of relapsing infections, which can go on to cause illness, death, and obviously sustained transmission. Radical cure of PVivax can be achieved by anti-hypnozoids like primaquine. The challenge with primaquine treatment is that it causes hemolysis in individuals who are deficient for glucose-6 dehydrogenase enzyme. G6PD testing therefore needs to be done prior to prescribing primaquine. To minimize risk of hemolysis, primaquine is often given at the low doses over a prolonged period of time, but the challenge with that is the poor treatment adherence. In PNG, effective control of PVivax has been hindered by a lack of G6PD testing and effective primaquine treatment. So in this work, a revised case management strategy for patients with symptomatic PVivax malaria who present to health facilities will be implemented. The key questions will be around the operational feasibility and the cost effectiveness of this intervention and the pharmacovigilance system. This program of work will provide evidence to facilitate widespread adoption of the improved case management tools and processes for safe and effective radical cure of patients with PVivax malaria, both in PNG and throughout other Vivax endemic countries. Lastly, IMR with National Malaria Control Program, provincial health authorities and other partners will conduct monitoring and evaluation of the HMM program that is currently being implemented by the Rotarians Against Malaria. In this work, we will assess the outcome and impact of the program, acceptability and the cost effectiveness of this program. This work will provide the National Department of Health and the Global Fund with the evidence needed to inform decisions on the scale-up of the program and the continued funding support for sustainability of the HMM program in PNG. If the program is found to have a greater positive impact on health and that it is accepted and cost-effective, timely malaria treatment can be brought closer to home and can reach a wider population, hence saving more lives. With that, thank you.
That was a brilliant session. Thank you so much, Dr. Laman and also Dr. Maria Ome, who's actually online at the moment. And Maria, there's a question that's just come through on the chat function. Jason Wow said quite extensive work being undertaken under various local shortcomings. What is the current state of preventative measures undertaken?
Thank you, Pamela, for the question. Well, from Jason, that is. Sorry, guys, I have bad internet connection, so. But from what I can hear, I'm assuming that the question was about current state of preventative measures in PNG, right? That point of view led by Mr. Leo Makita, so he'll comment on that chat, who's probably going to make a better comment on that. But we have that, and we have the IPTP, which for the pregnant women. Yeah, and so they're currently, we have those control measures that are in place, but at IMR, with vector-borne unit, we need to find out more more of the control measures that can improve, as like we highlighted in our research, in our session here. Malaria is difficult to eliminate. And for PNG, where we have vivax, it's a challenging species to deal with. And so there is need for some of the programs which I have mentioned in my concluding slides, the PAVE and the NATNA projects. They are the ones that are going to help us to inform our decisions on further control measures going forward for the country. Thank you.
Thank you, Maria.
Okay. I just had a bad connection. I thought none of that came across. So.
No, we heard you loud and clear, and thank you for persevering with us. We're going to move now on to our next spotlight, which is on Rebecca Emori, who's a research coordinator and PhD scholar with the PNG CHESS program. And after Rebecca's spotlight, we're going to go into our fourth session, which is on innovations in prevention of serious infections in infants. And that's going to be chaired by Dr. Anita Vanden Bigler. So stay tuned for this upcoming spotlight and session.
My name is Rebecca Emori. I work as a senior scientific officer with the Population Health and Demographic Unit. I currently work for the Comprehensive Health and Epidemiological Surveillance System, or CHESS, research project funded by the government of PNG. Moreover, I have been working as the main site manager for the CHESS research project since 2018. My research journey started back in 2006. From 2006 to 2021, I have worked and continue to work in different public health areas such as HIV, TB, maternal health, child health, population health, and demographic research. Health research has become my passion, and I have continued to develop my career in that area. Having gained valuable research skills, knowledge, and experiences in important public health issues, I have been able to win 2 Australian government scholarships, which I am grateful about. In 2013, I graduated with a master's degree from the University of Sydney in Australia. In 2020, I won my second Australia government scholarship to do a PhD program at the University of Wollongong in Australia. I am currently doing my PhD project or program at the University of Wollongong under the supervision of Professor Tony O'Clair, Dr. Anne-Marie Parrish, Dr. Tim Ha, and Dr. Vengiau. My PhD project is on translating child health data into policy and practice in PNG. This project will address key challenges faced by children in PNG, mainly children under 5. PNG continues to face significant child health problems despite having health policies, strategies, and plans in place. Importantly, PNGIMR produces child health and other public health data for the country, and evidence-based policy making is significant because it produces higher quality policies. However, the challenge is translating the research findings into appropriate health policies that the government can use to transform the lives of the children of PNG. This is where my PhD project will investigate and address. This research will use the child health data from the PNGIMR health surveillance system. It will further analyze the available PNG health policies related to children to create a homegrown health policy solution that will address priority child health agendas of PNG government. My PhD project will work with these important people and organizations: the Australia Award Scholarship, University of Wollongong, PNGIMR, and the government of PNG. I hope to survive in the COVID-19 era and pass my first year candidature in 2022, which will be a milestone for my PhD program. In future, I aspire to further develop my health policy skills so that I can contribute meaningfully and positively to PNGIMR and the government of PNG. Thank you.
Oh, good day, everyone. Greetings from Perth.
This last session on partnering for research impact may maybe take a little bit of a different angle in partnerships. We will share how the infectious diseases and immunity units at IMR and partners are integrating rather novel tools, state-of-the-art tools to study mechanistic pathways into clinical research to improve the health, the child health in PNG. As Geraldine Masiria will present, PNGIMR is now one of the very few research institutes in the world where systems immunology studies can be conducted in very young newborns, as young as 1 day old. And this impressive piece of work was developed in the last 5 years as part of the HIP-C EPIC consortium, a partnership between global omics experts and vaccine immunologists led by Ofer Levy from the Boston Children's Hospital. Now, the ability to conduct systems immunology studies at IMR has already helped us to secure funding from the Bill and Melinda Gates Foundations for the IMPROVE study that, as will be explained by Farhad Imam, is part of a larger Gates program of emerging strategies for infant probiotics to direct infants towards healthy early development pathways. Now, in the IMPROVE study, we hope to show that the use of probiotics in infants can reduce serious infections that, as Dr. Kasparia, a pediatrician at Goroka Hospital, will present are most common in PNG infants. And we will finish the session with Joycelyn Sapura, who will conclude with her and her team's experience with the probiotics trial. And I hope you're going to enjoy the session. See you later.
My name is Geraldine Masire, and I will be presenting on the world of omics. Young babies in Papua New Guinea are at high risk of developing and dying from serious infectious illnesses in the first 6 months of life. New technologies called the systems biology or the omics can now analyze complex interactions within the biological system and have made it possible to study the early development of a baby's immune system using small amounts of blood. This was shown in pilot studies conducted here in Papua New Guinea and in the Gambia. A training at the MRC unit, the Gambia, was conducted in April 2019. My colleague Sarah Javati and I had the privilege of traveling to and witnessed firsthand of how the study was being conducted in the Gambia and how the clinical and laboratory methods could be implemented in the PNG EPIC Omics study. As you can see, these are some pictures of the training that was conducted in the Gambia. The EPIC Omics study began in PNG in mid-year 2020. Pregnant women have been assented at the antenatal clinic and at the labor ward at Goroka Base Hospital. Assented women are approached within 24 hours of delivery to confirm consent and enrollment, if eligible. Babies are then randomized into one of 4 groups. To study immune development in the first week of life in the absence or presence of different neonatal vaccines and how this influences vaccine outcomes. Vaccines are delayed in some groups, as you can see on the slide. To harmonize vaccine schedules in PNG and the Gambia, children are vaccinated at 6, 10, and 14 weeks instead of 1, 2, and 3 months, and receive 2 doses of rotavirus vaccines, which is actually been given for the first time here in PNG. The team involved with working on the EPIC Omics study have faced a few challenges as the study commenced and progressed. These challenges were encountered during recruitment and field follow-up. The clinical challenges faced are recruitment from antenatal clinic, language or communication barriers between mothers and hospital staff, resulting in babies being vaccinated at birth, deeming them ineligible for enrollment. Sample collection, especially in newborns, is at times difficult, and the required amount of blood volume is not always obtained. Delaying the vaccines from 1, 2, and 3 months to 6 10 and 14 weeks has caused confusion and anxiety in a few of mothers of infant participants. Field challenges are geographical barriers, road conditions at times make participant locations inaccessible, thus participants' follow-ups are not always conducted. A few locations are also too remote to access by car or by walking, which results in missed follow-up visits. Migration, participants move from place to place due to personal, customary, and political reasons. Community misconceptions of vaccines. Quite recently, this is due to the COVID-19 pandemic. This is just a picture of community awareness being conducted in the communities. Samples collected in PNG will be sent to laboratories across the world for analysis. Together with the samples collected from newborns in the Gambia, the aim is to identify biomarkers predicting good or poor vaccine outcomes when given to newborns in low-income settings. The expectation is that these findings can be used to help develop vaccines that can be given to babies early in life and lead to good protection in all. Other outcomes and benefits of this study is that we have demonstrated that we can collect very small amounts of blood from newborns in our setting in PNG, and that these samples are of high quality, such they can be analyzed by our collaborators using omics technologies to generate big data that we can then use to study the immunology and biology of vaccine responses, but also diseases in a way that was not possible before. This has positioned PNGIMR as one of the very few research institutes that can do these kinds of studies in a low-income setting, and has already resulted in one other study at the IMR called the IMPROVE study. We hope this may attract the interest of current and potential collaborators to partner with IMR. To conclude, I would like to acknowledge the EPIC-HIPC study team, the clinical and lab team, for their contribution in this study. The study receives funding from the NIH. Thank you.
Welcome all. I'm delighted to participate in today's seminar from the Institute of Medical Research in Papua New Guinea, focused on partnering for research impact, and specifically on the session of emerging strategies for infant probiotics. My name is Farhad Imam, and I'm a neonatologist, pediatrician, and biochemist. At the Bill and Melinda Gates Foundation, we try and look at the intersection of where the areas of greatest need are and how they combine with areas that we can have the most impact. On my team, titled Maternal Newborn Child Health Discovery and Tools, we have a perspective of innovation that we bring to a landscape of available and emerging tools. Specifically, the focus of today's talk is going to be probiotics in the newborn space. This comes from a background of resiliency and growth of resiliency framework, which we've adopted. As we've been learning from data coming in, infants can die from pneumonia or diarrhea, but often these infants are vulnerable to begin with, born underweight or with growth faltering or wasting. And so we want to identify those infants who are most vulnerable, try and intervene as early as possible in the life course, and do this in an iterative way where we learn from each new study, each new intervention, and can continue to get better at trying to support these vulnerable populations. And what you see here is a depiction of a healthy maternal and newborn birth and infancy experience contrasted with that of a vulnerable trajectory of potentially an anemic or malnourished mom and an infant who encounters the same pathogen challenges but is unable to come back to the same level of health from each one and eventually can succumb at any of several time points. What we've learned so far about growth faltering is that probiotics can not only decrease inflammation, as you see here by measure inflammatory markers on the stool, but also can have benefits for growth. This was a pilot study which was not powered for growth, and yet the results of probiotic administration were strong enough that they showed up in the findings. And now we are doing a larger validation study of probiotics for growth in infants with wasting, such as hospitalized infants with severe acute malnutrition. This is piggybacked on top of prior results from a study in the low resource setting based in India, where probiotics supplied to newborns were able to prevent episodes of clinical or culture positive sepsis in newborns at quite dramatic levels, 40% or 80% respectively. These two different probiotics for growth and for sepsis prophylaxis have now been incorporated into ongoing studies in the portfolio that we manage, and combined with cutting edge tools for biomarker discovery, such as illustrated here. On the left, you see an untargeted metabolomic platform. So this is a method to measure thousands of metabolites in one drop of blood or in a drop of stool. For example, we have been able to use this in COVID to identify prospectively risks of hospitalization or death at the time of admission that outstrip or outperform the clinical indicators that we know are very powerful, such as age, heart disease, and other underlying risk factors that are known. So if we take that and we apply that to the nutrition space, to the microbiome space, there's a huge potential for discovery of novel biomarkers, novel mechanisms. And if you combine that with the ability on the right to do metagenomic sequencing and get a sense for what all the different microbial strains are, if you combine the strains with how the strains interact with food and create secondary metabolites, all of a sudden we start to get a much more complex insight and understanding into what the states of health and disease are. Now, there are two very important studies that are supported by the Foundation that I'd like to focus on today, one of which is well known to you. This is the intervention in Papua New Guinea, or IMPROVE study, hosted out of Western Australia, Telethon Kids, as well as your IMR. And you can see that there is not only two different probiotic strains, a Lactobacillus plantarum and a Bifidobacterium infantis, in interventional arms, in contrast to placebo, but there's also a rich and detailed biospecimen collection plan here. And so this is, again, a study that's not only important to show safety and tolerability of two potentially very important and impactful microbial interventions, but also is going to, while it's occurring, gather biosamples with the potential for discovery of novel mechanism, novel insight, and novel biomarkers into these to better inform future treatments and future studies. So we're quite excited about this study, as well as its combination with another study based in Dhaka, Bangladesh, which is doing something very similar. Also looking at these two different strains, also including high density sampling with this untargeted discovery perspective. And they are also structuring themselves quite similarly in terms of the cohorts of kids that are going to be followed up. That is going to be a larger study, and the two together we think are going to be an incredibly informative group of data to help inform us going forward on both growth as well as sepsis prophylaxis and inflammation in this vulnerable neonatal population. So taking a step back and thinking about what our greater goals are and how this fits in, what we really want to be able to do is to zoom in and focus in on this high risk group of newborns, which you see here depicted in, let's say, the red portion of the circle to the left. And we recognize that those are not all the same newborn with all the same risks, but rather a compilation of multiple different vulnerabilities, which may have an underlying shared vulnerable underpinning, as we discussed before, but ultimately which will be impacted and succumb to different diseases. And so here we want to make sure that we have effective tools that are able to address wasting, sepsis, low birth weight babies babies who suffer from prematurity. And so in terms of making sure that we have the best toolbox possible to complement all of the different other tools that are already available, what are the best new things that we can bring to the table? So I hope that I've been able to give you a little glimpse into that and into the foundation strategy and into your key role in this strategy with the improved study. So thank you for your attention. Delighted for the opportunity to give this vignette with you all, and looking very much forward to the work that we're already doing and the data and knowledge that it will produce. Thank you.
Good morning. My name is Castaria Mond. I'm a pediatrician at the Goroka Provincial Hospital. This morning, I will be talking about the common illnesses that we see at our hospital, as well as my thoughts on the potential impacts of probiotics. This is the outline of my presentation. To the pediatric ward, 1,500 children and thereabouts are admitted every year for the last 3 years. We admit children who are 8 days old to 13 years of age, and the common admissions are pneumonia, severe malnutrition, tuberculosis, gastroenteritis, and meningitis. To the special care nursery, we have cases of prematurity and low birth weight being the most common cause of admission, as well as birth asphyxia, neonatal sepsis, and risk of sepsis, as well as meconium aspiration syndrome. A risk of sepsis are those babies who are born in the villages outside of hospital or whose mothers have had prolonged rupture of membranes. In the brackets are the total percentages showing the percentage of admissions, total admissions for these diagnoses. In the nursery, about 700 to 800 children, neonates, are admitted every year. Neonatal infections, or what we call neonatal sepsis, make up a large proportion of admissions to the special care nursery. As you can see, between close to 200 to 300 children are admitted every year. And we have deaths that give us a case fatality between 9 to 14%. In 2020, deaths were increased because we had very sick babies coming in and fewer admissions because of COVID restrictions in travel and lack of access to health facilities. That's a possibility for why the case fatality rate was much higher than the other years. Pneumonia cases in the pediatric ward are the leading cause of admissions. Between almost 600 to 700 cases are admitted every year. And every year for the last 3 years, we have had 22 deaths. Case fatalities range from 3.3% to 4%. A study in India showed benefits in infants who receive probiotics. Some of these infants, their conditions are similar to those of children in PNG, as we have high rates of neonatal infection, there is pneumonia in PNG, and meningitis. None of the babies in the current study that IMR is doing has required admission to the hospital for severe illness so far. So hopefully this pilot study shows that probiotics that are given are beneficial, so that a larger study may be done in the future. And sometime down the future, if these probiotics are shown to be beneficial for infants, then it may become part of standard treatment for children to help reduce further the high rates of infections that we have in our infants. Thank you.
This presentation is on IMPROVE study. impact of probiotic intervention that is currently ongoing in PNG, conducted by PNGIMR and collaborators. Probiotics are harmless, live harmless good bacteria that can have significant health benefits in adults and children. A study in India showed that 7 days of probiotic supplementation starting on day 2 to 4 of life reduced early life infections, including neonatal sepsis, diarrhea, and pneumonia infections. Probiotic supplementation has not been studied and is not used in PNG infants, but could be a simple, safe, affordable, and beneficial intervention to prevent serious infections and deaths in infants. The study is a double-blinded, randomized controlled trial of probiotic supplementation in PNG newborns. The primary objective is to demonstrate the feasibility and tolerability of giving PNG infants probiotic supplementation for 7 consecutive days, starting when the infant is less than 72 hours old. Children are randomized 1:1:1 to receive one of 2 symbiotic combinations or placebo. The intervention is diluted with 3 ml of either breast milk or sterile water and is administered in drops using a pipette, as can be seen from this short video by my colleague Birenu. The different specimens are collected at different time points. Routine infant vaccines are also given. Rectal swabs and stools will be analyzed by PCR at PNGIMR for the persistence and presence of probiotic strains in the gut. Culture for bacterial carriage from nasopharyngeal swabs are also done at will also be done at PNGIMR. Small volumes of blood are collected at the start and end of the intervention using the systems biology approach as first introduced and mentioned by EPIC HIPSI study. The photographs shown here are of the improved study team at work collecting specimen and intervention being administered. The benefits and challenges of administering the intervention. The benefits include all newborns have well tolerated the intervention so far. There have been no severe adverse events. No infants were hospitalized for infections like sepsis or pneumonia so far. The infants are constantly gaining weight and quicker than usual for physical development as observed by clinician and parent. The challenges faced: most times the newborns sleep soundly and have to be woken up to administer the intervention. Mothers sometimes fear that their babies might die in their sleep because they sleep a lot. Some communities have a misconception that participants are sick. The improved study team perspective is that there are many challenges in actually implementing the study, and the challenges faced, they vary each day. However, the intervention seems to have health benefits, and if we can confirm that in the ongoing study, this would be a good intervention that parents can give themselves to their babies. With that, I would like to acknowledge the improved study team, and this study receives funding from Imprint and Bill and Melinda Gates Foundation. Thank you.
So hello again. I just would like to share this acknowledgement slide because I think you can never acknowledge all the people that make this happen enough. A special thank you as well to Jocelyn and Geraldine, which I believe were not even able to watch the session because I understood that they are doing follow-up visits on the babies. I just wondered, you know, as well, if other people are interested in incorporating these kind of systems biology approaches into their studies, that we would be very excited to talk about it offline. Now, before we move on, I was asked this morning by the Westphalian Center of Vaccines and Infectious Diseases at the Telton Kids Institute to share that since 2019, the Westphalian Center has had the Deborah Lehman Research Award, which is say up to a 30,000 Australian dollars award for training and or development of an early mid-career or higher degree research student. I have not been given information when is the next deadline for the 2022 award, but I understand that this will be up soon. So for any people who are interested and would like more information, please email wisfit@teltonkids. Thank you. If there's any questions, I'll take them, but I understand it's all okay.
Thank you so much, Anita. Thank you for leading such a great session. Coming up next, we have 2 spotlights that we want to share with you. And after that, we're going to cut to a live panel with our partners. So please stay tuned for those. Our first spotlight will feature Dr. John Bolgna, who actually was a panel member in sessions 2. So he's going to join us from Madang. And the second spotlight is one of our senior research fellows, Dr. Celestine Aho.
I am John Bolgna, a specialist obstetrician and gynecologist from Madang Provincial Hospital, who has had more than 11 years of research collaboration and partnership with the Institute of Medical Research and the international collaborators, and also some local partners as well. I see partnering for research impact is paramount in that it shapes the way we practice medicine. Its health impacts are many, and I have been fortunate to have partnered with local and international collaborators here in Madang in some of the very important research agendas that have actually made big impacts in Papua New Guinea. For example, the HIPTP study that we looked at Fancida, which is now the normal prophylactic treatment for antenatal care women, taking the place of chloroquine, which developed a lot of resistance. So that's actually changed the way we look after women folks in the antenatal care in this country. And the other one is a misoprostol clinical trial that I have led with Dr. Moses Laman in looking at how induction is performed and getting the clinical protocol right for our country. And that has changed, really made a big impact in the way we induce women who have gone overdue, or if a woman is a risk factor, or the baby needs to be delivered. So that's actually changed the way clinical practice has been done in obstetrics and gynecology in this country. Also, in partnership, we are able to create infrastructure and some logistics support and training, especially for clinicians in the hospital settings with research skills, research tools, which are required for professional development and also for career opportunities. So capacity building has been one of the mainstay in what in our partnership with research collaborators. And this has actually seen a lot of registrars and doctors and nurses being trained in understanding research agendas. During the 11 years that I have had collaborated both locally and internationally, we have conducted some of the biggest clinical trials. Some of them are like HIPTP with Stephen Rogerson, the one-time trial with Andrew Vallely, and HPV point of care testing and treatment with also Andrew Vallely, and also the misoprostol trial that was conducted, as I've mentioned earlier, with Dr. Moses Laman. And recently, the support trial with Holger Unger from the Menji School. There's been many more clinical research that have also been done and that are ongoing now. And I look forward to fostering this relationship and collaboration into the future. In partnering for research impact, although it may sound trivial to some people, winning research grants is also very, very important. It provides employment for our local people. And if it can provide employment and also be able to have bread and butter on the table and also put a child to school, then I look forward to continuing this relationship into the future. Also, there's been a lot of career opportunities in training with research, partnering with research collaborators. A lot of registrars, medical registrars, as well as some nurses, they are able to do postgraduate studies that they finally had their research projects published in internationally peer-reviewed journals. And some have actually built their career interest in research and have gone on to become researchers, and some have actually progressed on with the postgraduate studies and have become leaders and opinion leaders in the country. So there's a lot of educational opportunities when we look at partnering with research as well in this agenda. There have been many clinicians who have, as I said, have taken educational opportunities, particularly in child health and obstetrics and gynecology, in the field at which I am involved in. Many registrars have completed diplomas and masters. We have been able to get assistance with the research partners to actually get those papers published. Some have become specialists now. I have just completed my medical doctorate and just graduated last week. And there is a lot of benefit, both directly and indirectly. We need to, in looking at research partnership and its impact, that we need to take stock of the practice that we do and contribute to building an evidence-based practice in our clinical setting locally. And that is very important. I mean, research alone actually is able to change practice. Some practice of the past may not be the same practice of the future. We need to see what is locally important and make changes to our clinical practice guidelines. And that we need to partner with research bodies to enable changes that suit our requirements. But this comes with a lot of work and sacrifice. Just being a clinician is not enough. We need to collaborate with major partners and this involves a lot of commitment. Sometimes I find that we need to sensitize the nurses and everyone else that we work with to research culture, and that is difficult. And also we may have some conflicts that need to be resolved with research collaborators and also locally. So we need to create a really conducive environment that we can work with our research partners and collaborators. So there's a lot of sacrifice and time needs to be spent, and we have to be committed. But again, it is very rewarding. We learn new things. We supervise students and then make them aware of research activities. And then some can even progress on to formal qualifications as a result. So to our international partners and collaborators, times have changed. Many young generation Papua New Guinean clinicians are now very aware of the important and the significance of having research skills and have to collaborate, and they are willing to participate. But at the same time, we have to also have mutual benefit for the research agenda, as well as for positive capacity building, and put everyone's interest at first. And then we can progress this and make true impact in partnering into the future. Thank you.
My postdoc research is on middle ear infection, also known as otitis media. Otitis media, or OM, is an important cause of hearing loss in children and adults. Hearing loss in children can have a negative impact on their language development and comprehension, and this in turn can affect their education and their job prospects as adults. Understanding the OM burden of a population and the microbial causes of OM is essential for developing strategies for early and effective prevention of OM onset and to stop ear disease progression. We know little about OM and hearing loss in PNG. This month, we will recruit participants in a study to investigate OM in a cross-section of children attending urban outpatient clinics in Goroka. And this is to, one, find how common OM is in children and why some children are more likely to get OM than others, and two, to establish the microbial causes of discharging ears. This is a collaboration between the IMR, the Eastern Highlands Provincial Hospital, Telethon Kids Institute in Perth, and the Menzies School of Health Research in Darwin. Through the study, we plan to investigate the use of telehealth, investigate how support and diagnosis can be done remotely. The study is funded by the West Farmer Centre of Vaccine and Infectious Disease Deborah Lehman Research Award, which I was a recipient of in 2018. And recently, we got a small grant from the Centre of Research Excellence in Ear and Hearing Health for Aboriginal and Torres Strait Islander children, which will support the laboratory work. My interest in OM research comes from having a personal experience with my younger brother having chronic OM as a child and then experiencing moderate hearing loss as an adult. My career journey at the IMR would not have been possible without IMR's partners in health research. I joined the IMR in the middle of 2006 after uni and worked on a 7-valent pneumococcal conjugate vaccine trial. I did my honours work with Dr. Andrew Greenhill, then the bacteriology head, through the Fogarty program, which became available through the collaboration between Case Western University in Ohio and the IMR. I then went on to do a Master's in Science in 2010 at the University of Basel through the partnership between the Swiss Tropical Health Institute and the IMR, and did my research work under Professor Gert Puschke's laboratory. In 2015, I went to Darwin to do a PhD with the eHealth group at the Menzies School of Health Research under Associate Professor Heidi von Smith's lab. Through my journey, I've had a number of national researchers and our collaborative scientists and researchers who aided me along and inspired me to do the work that I do. My research journey would not have been possible without IMR's partners and support.
Well, those are the final spotlights. I hope you've enjoyed tuning in and hearing the stories, the challenges, as well as the victories that we face every day on the research field here in Papua New Guinea. We're going to move on now to our final session of the symposium, and that is a panel discussion with some of our partners. We're so fortunate this morning or today to be joined by Professor Michael Alpers from Curtin University, who is one of our former directors of the Institute. We're also joined today by Anna Malson, who is the acting WHO representative in Papua New Guinea. We're joined by Dr. Manuel Hetzel from the Swiss Tropical and Public Health Institute in Switzerland, and also Professor Leanne Robinson from the Burnett Institute and WEHI as well, I think. Is that correct, Leanne? Thank you. So we're going to give everyone an opportunity to just share their reflections on this symposium that we've just been a part of, and the presentations and the sharings, and then get them to also speak on their reflections on the partnerships that they've kind of worked in as part of partners at the Institute, and as well as when they worked with the Institute. So we might start with Professor Alpers.
Oh, well, thank you. Right. Yeah, I have to be brief, of course. So I thought I'd talk about congratulations, decolonization, buttressing, business, and community. Okay, congratulations. Well, I can't believe it, really. Congratulations, Willie, for your leadership and to everyone for their presentations. It was just awesome. I'm so moved, I must say. You are just wonderful. So it's a vision made into reality, and I'm just so happy. So congratulations all. The world is going through a process of decolonization at the moment, and you'll find it everywhere in all kinds of contexts, including medical research institutes. And I must say that PNGIMR decolonized in 1977, so that we were well ahead of the game. And that was done through support, strong support from the government to a national institution, gradual build-up of a strong cadre of Papua New Guinean scientists. And what's happened in recent years is this is sort of exponentially increased. It's just amazing. And colleagues from overseas who came to work, and partnerships that were built with institutions and colleagues nationally and internationally. And that was a great success. I must say, not all the colleagues and partnerships were anti-colonial, and there's some good stories I could tell about some of the problems we had. But when I left, we incorporated this concept into the Buttressing Coalition. And I think it's pretty clear what that means. And if anyone asks, what does the Buttressing Coalition mean? Well, you know then you have a little educating to do. And it's so pleasing to see that it's still flourishing. So that's really talking about the past and the present, and all these strong partnerships that are present at the moment gives us great hope for the future. For the future, I had two words, business and community. And when I left, one of the things I thought we should do was to expand the contribution of the business community within PNG towards funding the institute and medical research. And you'll find that this is, well, standard throughout the world now. And I wanted to increase the council of the institute and bring in some business members who would contribute expertise, but also we hoped funding and seek funding elsewhere within the business community within PNG. And Peter Barter thought this was a great idea. But then when they did change the council, instead of expanding it, it was contracted. And I was very disappointed by that. And I think this is something that should still be borne in mind. I think we should have at least a few members on the council from the PNG business community. And finally, community. Well, community is the basis of all our work. It always has been and always will be. But I think we need to build the demand side for good health, as well as the supply side. And many communities are not demanding good health because they don't understand it and they don't see the possibility of much health improvement. And we need to try and build that demand. And we can do that, I think, through collaborations. And I was so pleased to see how well the collaboration between the health services and PNGIMR and between PNGIMR and NGOs are at the moment. That's really encouraging. So I think we need to build this collaboration, health services, NGOs, and the community. And it needs to be done on a broad basis. So I think we need a unit that will draw together all the various disease-based studies into health promotion, health improvement within the community with the involvement of communities. And that's something that I could just put up as a suggestion at this stage. Okay, so I've come to the end, and I look forward to hearing what all the others have to say. And congratulations to you too, Pamela. You are an MC.
Thank you so much, Michael. It's so lovely to see you, and thank you for those comments. Thank you so much. Anna, could we go to you to share your reflections on partnerships? We've had a lot to do with you, obviously, recently, and with WHO, with the COVID pandemic and the response, the country's response. So please, the floor is yours.
Thank you, Pam. Good afternoon, good morning, everyone, wherever you are. It's a real honor to be here today on this panel on behalf of the World Health Organization for this virtual symposium. And I think we can all agree that the Institute is a national treasure, and there are a few countries of similar economies that can boast having a homegrown National Research Institute of the caliber of IMR. So I iterate there, Prof's congratulations to the Institute. As we're all together living through these unprecedented times in the COVID-19 pandemic, there has been no time like this where partnerships and research have mattered more, not only globally, but also locally. And to do this, we need to work closely with the sector, the health sector, and sectors beyond the health, using a whole of government, whole of society approach to promote health and address risk factors that drive the epidemic. The pandemic has brought us an important opportunity to partner differently, to work together differently, and drive the need for faster, more accurate data and research to inform decision-making on the run. Continuing to build the partnership between IMR and WHO and leaders that influence policy making at the highest level will be essential. Partnerships and collaborations will be crucial to our success, even more so in the light that the fiscal pressures that will come as a result of the contraction of the global economy, transition from donor financing will bring. Operational and implementation research will be critical in producing valuable evidence to assist in making health services more efficient, interconnected, sustainable, and to reach everyone that needs them. Responding to the pandemic in PNG has shown us how important local context matters. Whether it's trying to get information across about the virus, how to adopt the new normal, Nuplapassin, or the benefits of vaccination and addressing hesitancy, PNG has shown us that we need more nuanced information. And WHO is pleased to be collaborating with IMR, UNSW, and the Kirby Institute to research the impacts of the response to COVID-19 within the primary healthcare system and infectious disease programs as part of our research development agenda. This also now brings me to my second theme, new ways to work at country level to produce results and improve health indicators. The Counter Me Too study demonstrated how research can be creatively leveraged to increase coverage and access testing and treatment. By strengthening health systems, WHO and the IMR are working with the Department of Health to address critical bottlenecks and access life-saving interventions. The challenges that PNG faces in the delivery of primary healthcare is reflected in the current response to COVID-19 and the vaccine-derived polio outbreak. It is unacceptable that some districts' immunization coverage is as low as 40%, and yet we have the vaccines and the technology to be able to ensure that children are protected. What we are learning, however, from the COVID-19 response is that by working together, we can reach more people more efficiently. Translating this experience to a sustained integrated service delivery model with basic health services is something that requires much work. When WHO was founded, communicable diseases were our chief concern, most like the IMR. And collectively, we continue to make strong progress in our fight against these deadly diseases. The work of IMR in malaria control has been instrumental to the recent success of the National Malaria Control Programme. And whilst many challenges lay ahead, we have some of the richest local research to continue to inform and develop the programme. New partnerships are emerging out of malaria control, such as STRIVE, bringing new and old partners together, IMR, Burnett, WEHI and others, and different expertise to support and influence the programme. IMR continues to play a central role in the fight against malaria in Papua New Guinea. But there are also other programs of significant work in disease control. The seminal work of the IBBS, for example, gives us new and critically important information to ensure that key populations are able to access services without discrimination or judgment. In signing up to the SDGs, PNG has made a commitment to leave no one behind, and the Counter Me Too research provides us the information that we need to reorient our health services to be more people-centered. IMR has played an instrumental part in multi-drug resistant TB, along with research into other antimicrobial resistant pathogens. Keeping our fingers on the pulse is critical, as this is one of the most frightening threats in the areas that we work. And this brings me to our final way of working together, ensuring that we leverage each other's strengths to safeguard global and national health security. WHO, through its Health Emergencies Programme, has a clear mission to protect and save lives during outbreak and emergencies. IMR, through its influenza and other outbreak surveillance work, is a vital part of the development of PNG's core capacities to respond to outbreaks and emergencies. It was because of this expertise and its institutional partnerships that IMR was quickly able to pivot to set up the scaling of testing for SARS-CoV-2. We should all reflect and celebrate. This is one of the key achievements of the successes of PNG's COVID response so far. We need to continue to strengthen the Institute's expertise and hard work in many of these areas. The continued need for monitoring and surveillance as part of this is again a way that we can continue to work together to build more resilient health systems. This has been an incredible opportunity for IMR to cement its place as instrumental to health security in PNG and provides a platform for us to continue to strengthen partnerships for health. In looking forward, we see that there's a considering the global agenda and expanded need for health research that not only focuses on the medical, but public health and the determinants of health. IMR is well placed to continue to support the growing agenda. But collectively, we need to continue to strive to make best use of the wealth of the evidence and information that is produced by IMR, strengthening the linkages to policy and planning, and developing mechanisms that allow greater sharing of information and platforms for policy dialogue. Back in February 2020, I had the pleasure to support IMR in its strategic planning exercise, and contained in there was a vision so clear for the future that it's even more relevant today. And that IMR has the purpose to lead and accelerate innovation to improve the health and well-being of populations through research and implementation science. And to do this by addressing the intersection between communities, research, health systems, and policy. WHO is proud of our long partnership and collaborative work with the Institute, and we can take comfort in the fact together that we have begun to develop the foundations for future success. We look forward to continuing to grow and develop our linkages, and we look to IMR to continue to provide the evidence for research that not only informs local policy guidelines and plannings, but continues to contribute globally. Thanks, Pam.
Thank you, Anna. Thank you for your continued support and support of WHO. We might cross to Manuel. It's very early in the morning in Switzerland, but we're so glad that you've joined us to share your reflections on these important partnerships.
Thank you so much, Pam. And indeed, it really is a pleasure to be part of this panel. It's an honor to be here with you this morning. It wasn't a problem for me getting up at all, a little bit early to be part of this. And again, also from my side, really congratulations to you for organizing this event. I think it's amazing to see how fast we have all moved in this new virtual world as well, with Zoom and all these other applications bringing us so close together. I almost feel like I'm actually sitting in Goroka in the library with you. Unfortunately, I'm not, and probably there's one thing that we can never change, that's the time difference. But nevertheless, I mean, I think having the panel of partners here and also seeing this symposium really shows us already how important partnership is. And I must say, from my very own personal experience, it's a unique experience working together with IMR, and having been in Goroka for a number of years, and now unfortunately physically quite far away, I still feel very much part of the IMR family. And this family spirit, I think, is something that is very unique, and that is something that reflects a long-term partnership. You know, you're always part of your family, so it's very difficult to leave, and you don't want to leave because your family makes you feel at home. And I think the long-term partnerships is something that IMR has really managed to build, and is probably the core of what brings IMR and all of us together forward with research in PNG, and trying to implement, or trying to inform the implementation of evidence-based health decision-making in PNG as well. So I think one thing I want to briefly speak about is the recognition and appreciation of IMR. What I think we can all agree with is that IMR is an institution that is actually well recognized globally, and that, you know, people have recognized in many countries around the world is unique in such a setting. However, sometimes I've wondered whether IMR is appreciated within PNG as much as it is appreciated outside of PNG. And I think this appreciation that I feel we do see very often when we go into communities, and people are happy to see IMR, you know, enter the communities and work with the communities, as Michael Alpes has pointed out so correctly, the importance of working with communities. I think it is very important that this partnership and recognition is also there from a government level. And I think it's been, in my own work with IMR, it's been very encouraging to see how well appreciated IMR's input is into the National Malaria Control Program, and the close collaborations that we've had together with other partners, Burnett Institute, WeHi, and others, informing and working really closely with the stakeholders at NDOH, with NGOs for the malaria program. But how is that further up the hierarchy? Do we really have this appreciation? And this brings me to the question, sometimes maybe we undermarket ourselves a little bit as IMR. Is the Institute really self-confident enough to say, you know, this is what we have to offer, and we really have something important to say because we know what we're talking about? And this, I think, is also, you know, it's important to consider in this context that research is not something that can be done overnight. It's the partnerships that cannot be built overnight, and this is why these long-term partnerships that are obviously also reflected in Buttressing Coalition are important, but evidence cannot be generated overnight. It needs a long-term commitment to work, and it needs a long-term commitment to finance the institution, but also the research activities. And then the evidence that's generated, Michael Alpes talked about demand, and I demand for good health, but I also think it's important to have the demand for evidence to then build interventions based on evidence. So this translation of evidence into actionable practice, I think, is really something where IMR can also be stronger and can be more self-confident to inform this process. And I think, as what was really also well alluded to by Anna Marlson, is that the health situation or the burden of disease is evolving wherever we are. We have this epidemiological transition that we've seen across the world in many countries over time, you know, moving from a higher burden of infectious diseases to non-communicable diseases, which is certainly something that is very relevant in PNG. But the current COVID epidemic obviously also alerts us that communicable diseases aren't over across the world, and it's really something that we need to keep in mind, where we need to invest in surveillance, and where maybe, you know, also we can learn from the experience of IMR in surveillance activities and work very closely with the health department and other partners. And this maybe brings me to two last points, is that I think while we can all appreciate in a professional context the importance of generating evidence on disease burden, on the relevance, or on the effectiveness of interventions, maybe sometimes we need to be better in making also a business case about how important improving health is and how important generation of evidence for improving health is. And this entire area of cost effectiveness research maybe is something that in which we can maybe be stronger and also build new, stronger partnerships to provide this information. What is very clear, and also this was mentioned, the health evidence, the translation of evidence into action is something very context specific. And PNG, in my opinion, is really blessed to have IMR, an institution that is able to generate this local evidence that can inform policy. Obviously, being able to provide this evidence requires growing local capacity. And I'm very pleased, I must say, to see over the years how partnerships to strengthen capacity have really grown. And I think one of the biggest pleasures to see the number of Papua New Guinean local scientists, researchers who are currently undertaking advanced studies for a PhD or for a master's degree. This is something, when I joined IMR had a very small number of PhD holders, and now I can see so many young scientists in PhD degree programs. And it's something that you never or rarely actually read in the newspapers, what an incredible achievement of IMR that is. And that's really my last point. I want to congratulate IMR on this amazing achievement of building up this capacity and strengthening the local research capacity. And congratulations also to all the partners for supporting this, and also from our side, Swiss TPH, really to reiterate that we are very keen to support this capacity building whenever and wherever we are. So thank you very much from my side for now.
Thank you so much, Manuel. Thank you for just your continued support, the support of the Swiss Tropical Institute. Thank you for the challenges that you've thrown at us about possibly marketing ourselves better. I mean, this virtual symposium is hopefully the beginning of many where we can engage more widely with an audience in country as well as overseas. So thank you very much. And also your points about making a business case to actually put a value, like an actual dollar kina value, to what it'll cost if we do nothing, to make a case for support. So very valuable comments. We're going to cross now to Leanne, who joins us from Melbourne. Hello, Leanne. Thank you so much. And it'd be lovely to hear your reflections.
Thanks so much, Pam. And I mean, like the other panelists, it's just an honour to be on this panel and to join you all for this symposium. I just also want to say congratulations. Like, what an impressive and moving symposium this has been. I think just such a clear demonstration of how partnerships are essential to maximise the impact of research in Papua New Guinea. And I mean, just reflecting on the talks that we've heard today and just how clearly they've highlighted both the opportunities and the challenges of conducting innovative field and laboratory research in the PNG setting. And in particular, highlighting how all of this is only possible through the depth of local knowledge that has been built within IMR and partner organisations over decades of working with communities, with health facilities, with provincial and national health stakeholders in this way. And and the partnerships that people have, you know, their networks that make this work possible, as well as the reciprocal support, you know, required by all parties to really achieve the outcomes that we've seen in many of these talks. I think, you know, just like Manuel said, many of us that have had a unique privilege to work at IMR and really see firsthand what it takes to build and sustain these partnerships, to value and nurture them. I think it's just so gratifying, you know, to be able to see all this, just like what you said, Pam, really being displayed and advocated today. So just, yeah, really sort of warmed and proud and delighted to, you know, be so involved still. I think it's, you know, it's only by working in genuine partnership to conduct research that we really harness the diversity of skills and expertise that exists within all of our teams. And that includes the community. Like Michael said, I think the community have always and continue to play such an important role in our research and in even really informing, you know, what the research priorities are. But they also need to be heard. They need to be heard more. And through our research, I think there's that opportunity to advocate for the community, to create that demand that Anna talked about. I think, you know, by being willing to develop a shared vision for a project, I think we heard so many examples of where that's the case. We really promote equity, mutual benefit, courage, and openness. And all of these support effective partnerships and maximise the impact that we can have, because we leverage each other's key strengths, just like Anna said. And I think that's really how we can achieve the most impact together. It has been such a pleasure today to see all of PNGIMR's PhD and Master's graduates showcasing their achievements. You know, like Manuel, I mean, we were at the IMR at the same time, and, you know, part of the honours and master's programs when many people were doing them, and it's just so important that this body of PNG postdoctoral scientists continues to grow and lead the research programs of IMR, and I just want to really congratulate IMR on this. Almost to my last point, I think, you know, we've also heard numerous examples today of how working in this way, in partnership, ensures not only that the research being conducted prioritises the critical evidence gaps, but also that those research findings can very quickly have a direct impact on program implementation and policy. And I think we've seen that through numerous programs that have been talked about today, and therefore really address and have an impact on the health of all Papua New Guineans. So lastly, I'd just like to really agree with the points flagged by all other panelists in different ways, and yourself, Pam. I think that it's maybe time to be challenged, you know, to really advocate for IMR, like Manuel said, not only internationally, but also domestically, and to really use our research to advocate for better health for Papua New Guineans, to use our track record in conducting high quality, world-leading research for more funding, both domestically and internationally. So thanks again so much.
Thanks, Leanne. Thank you for everyone's initial reflections, very, very valuable comments. We have a little bit of time before we wrap up this panel, and I thought I'd use my discretion as the MC to just place a question to the panel, and it's then up to whoever wants to respond. Very recently, the PNGIMR has been challenged by the Prime Minister to do more research and development. While it's not in our mandate to do R&D, in your opinion, is this something that you see us pursuing, and what steps and considerations would need to be addressed if IMR wanted to rise to this challenge? Anyone like to go first, Michael?
Oh, well, research and development. I mean, I don't know how you can. Well, the research is certainly for the development of good health in the community of Papua New Guinea, all of whom the Prime Minister and his colleagues are meant to serve. And you cannot have wealth or well-being without good health. And if we're working towards that end, then we're certainly making a big contribution to the national estate.
Thanks, Michael. Can I invite one more response from the panel members before I invite Professor Pomat to make final concluding statements?
I'll say something, Pam, if you want. Yes, that was quite the challenge that was put out there for IMR. And that certainly comes from, a lot of the conversations that have happened through the COVID response here. Does the virus affect Papua New Guineans differently? Is our immunity different? How do we know this vaccine works in Papua New Guineans? So there's certainly a scope for looking at how we explore these questions in country. And I think having that local research to inform those discussions and to really show ownership around health in PNG is critically important. I think it's a hard decision to make because PNGIMR has been invested for so long in so much fantastic public health research. It's a very strong pivot, but there can always be a balance. And I think that was something that was talked about, back in the strategic planning. Where do we go with some of that? But I think that the IMR has such a clear role in public health in Papua New Guinea, and I'm a very passionate advocate for PNGIMR and a role in the National Public Health Institute model, whatever that may look like in PNG, and that's certainly back on the agenda. How do we consider a transition from a national control centre to developing a National Public Health Institute? And IMR should be front and centre and part of those conversations about how do we really get those essential public health functions working in country, extending that reach into districts down to through provincial health authorities. The outbreak and surveillance work that IMR is critical to as part of that. The development of professionals and scientists and that knowledge is so instrumental in the future here. And so I think it is a great challenge set by the Prime Minister, but also, again, I think it comes back to really selling what IMR does and what it does best, and supporting that information and that research to be used in the policy making and at the decision makers level. So being visible and being upfront, being in those conversations is critically important.
Thank you, Anna. And I guess as I mentioned earlier, we've really appreciated putting this symposium out there and moving to that virtual format. And I think it's the beginning of bigger things in terms of getting that research out and sharing the incredible work that the Institute does. I'm now going to invite our director, Professor Pomat, to come and make some concluding statements and remarks. Thank you again to the panel members for your contribution. Thank you for being part of this strong IMR family. It's absolutely been a pleasure to chat with you all and to see you all in the same room, even for a short amount of time. Professor Pomat.
Good afternoon, everyone, and good to see you all. Thank you, Manuel, for getting up so early in the morning to be part of this. And obviously, Michael and Deborah, all the way there in Perth. I hear there's a lot of rain there. I hope you're keeping safe. Anna, good to see you. And Leanne, I hope you are in Melbourne and not Newcastle. Yeah, so it's good to see you all. And Robin Davis sent his apology. He couldn't join the panel right now. But I'd like to say thank you to all of you for and for all the presentations that have gone ahead, all the partnerships that we have built over the years. And for Michael being especially the shoulder that a lot of us have stood on and made sure that the rigors of science were done in the way that we continue to do today. So thank you, Michael, for that. All of you mentioned family, the important partnerships that we have, and the long-standing partnership that this has continued to keep us going forward. And I think those are important for us. And I know that each of you are in many ways infected by the disease called Papua New Guinea, and probably the subspecies of that is PNGIMR. So thank you very much for continuing to be supportive of the Institute, for continuing to ensure that we drive the research agenda, but at the same time develop the infrastructures, the young scientists that are coming up. And I'm really proud that people like Pam have come back and are continuing to drive the agendas that we have. And we've got new people currently doing their PhDs. And yes, Michael, regarding the council, yes, Moses and I have spoken about the idea of going back to the Act, looking at the membership of the council so that we increase it to include business-oriented people who can drive the Institute in other ways or not in other ways, but drive it further in the ways that we are not currently at, because obviously being scientists, we're limited by our business knowledge. And I guess the only knowledge we have about business is whatever funds we receive and how do we report back and so on. So, but I'm really happy that all of you have come on the panel. All of you have accepted the challenge of time. Thank you, Manuel, again. And all of you have contributed to this panel to make it such a inspiring day for us at the Institute. And I think the partnership that we have built, and we haven't really shown everything. We've just shown some of it during the presentations that we had this morning. So I think we are all happy with that. And thank you to Anne Larson, who's behind the scenes somewhere in Western Australia, for getting this going, and Pam as well for agreeing to be the MC and making everything go well today. So thank you very much, everyone. We'll continue to be in contact with all of you for different things as we go on. Thank you, True.
Thank you, True.
And I will include as the thank you for your
Thank you so much. I just want to again thank everyone who dialed in to join us on this first ever virtual symposium of the PNGIMR. I can't mention everyone by name, but I do want to acknowledge the wider IMR family and partners who have worked so hard behind the scenes many weeks ago in advance to put together such an informative program to highlight the incredible work that we do. And as Professor Pomat mentioned, I do want to also acknowledge Anne Larson, who's been working behind the scenes all the way in Perth. So Anne, we do appreciate and acknowledge the hard work that you've put in. Thank you to the Buttressing Coalition who have funded this undertaking. We couldn't deliver this program virtually without their support. And I just want to conclude by saying that the motto of the PNGIMR is Save Tambuim Sik, and that means knowledge banishes illness. The research that we do generates knowledge, but it's also important that we share that knowledge to bring about lasting change. Thank you everyone for being part of this first ever virtual symposium. God bless you and take care.